Welcome to this third QNet workshop. As I said, we would like to welcome you all to this third edition. When we say we, well, this is the names behind the QNet coordination team, and all of them contributed to make this happen and to also carry on with this initiative. The topic of this third workshop is tropical QWA, or quantitative wood anatomy. Now, why that? Well, there is a couple of motivations behind choosing this topic. One is that the tropics are by far the most species-rich forest ecosystem, as we all know. And the tropics, they play an outstanding role as global carbon sinks. This is all very known to us, I'm sure. Still, and despite this importance, we have a huge knowledge gap in understanding tree growth in the tropics. So quantitative wood anatomy with tropical wood is limited by methodological constraints and resources. I think everyone who ever tried or did work with tropical woods knows very well what I'm talking about. And I'm also very sure that our speakers will highlight this a bit more in their presentations. And then last but not least, this was also a topic suggested by you. So this is just also to show you we basically are listening to your wishes, and we really try to serve you as a community. Together we can moving forward with quantitative wood anatomy. That's the goal here. So it should be interactive to the needs of you. Now the program of today's workshop, it's a bit a seminar style this time. You all got the invitation. So we have a introduction, about 15 minutes. And then we have 5 presentations by expert speakers. I don't read the titles here. However, everyone has roughly half an hour, of which maybe 10 minutes should be reserved for general discussions and questions. We also included a break, about 15 minutes. And then this is also very important, maybe when you remember the tentative program, the earliest program we sent out, we didn't include a general discussion. Now we really say, okay, let's also include like half an hour at the very end so that we can engage. Maybe you need time to think about the question, or maybe also when we put together everything, there is some overarching questions that pop up. So enough time for that. We are having one single session, so a very simple mode. As Angela already mentioned, the workshop is recorded, and we will also make it available through the QNet webpage in the membership area. Now, if you want to interact, you can do this anytime through the chat function. Please use this one, otherwise parts after every presentation or at the very end. So this you can do directly, but also we'll also monitor the chat and hopefully really enter the topic you probably use about the death of Dieter Eckstein. And here I would like to invite
to honor Dieter Eckstein, who recently passed away just about 2 weeks ago. Since I assume that none of his former PhD students are here, I take the opportunity just to say a few words to honor him. Dieter Eckstein was, I think, one of the founding fathers of dendrochronology, at least in the northern part of Germany. He studied wood science and technology in Hamburg University and finished his PhD thesis in 19 he continued to date music instruments and very famous in stimulating the development of dendroanatomy, or let's say the applications of wood anatomy in tree ring sciences. Dieter had many official positions. He was the dean of the wood biology department of the University of Hamburg, one of the very famous wood biology departments in Germany. He was acting as direction of the Federal Research Center for Forestry and Forest Products. This is a state. He was Director General of the Forest Research Center in Hamburg. But beside all of these official positions, Dieter is certainly being kept in memory as a very supportive person who stimulated tree ring science and also young talents in dendrochronology, and also serving for the whole tree ring community. So in these few highlights, I don't want to mention how many highly cited papers and publications and age factors he reached. I think it's more important to remind about the person, the warm-hearted person and mentor that Dieter was. So he served the community as the president of the International Tree Ring Society over 20 years. He was an acknowledged fellow of the International Academy of Wood Science, and he was also the initiator of the conference series Eurodendro that is still held today after more than, I think, 25 years. Finally, he was from the very first hour, one of the editorial board members of our very important journal for our community, Dendrochronologia, and also of tree ring research. I took all this information from an article that Ute Saas-Claessens, who was a PhD student of Dieter, wrote in 2005. The reason for that was the retirement of Dieter at Eurodendro 2004. If you could flip to the next slide, please, Georg. This is actually a slide that a colleague from China created, Yang Erian, one of his PhD students. And this is just to show you the international influence of Dieter, who was always supporting tree ring researchers in pioneer situations where they were among the first in their home countries to do wood anatomy studies or dendrochronological studies. So Dieter hosted PhD students from Eastern European countries, from Poland, from Lithuania, from Russia, but also from Southeast Asia, from Thailand, from Myanmar, and last but not least, also from China. So he's a very well-renowned person, especially in these Asian countries where he laid the very important foundations for the really rapid development of dendrochronology and wood anatomy. The result of it we see nowadays, and we could witness in a conference, in an online conference that organization. So we'll keep Dieter in a very honorable memory, and I thank you all for listening and for honoring him and his achievements. Thanks a lot.
Thank you very much, Achim. And I can only, well, say he's one of the big fathers in our field. Maybe an anniversary, celebrate the first year's anniversary. That is certainly nice, and we hope that there is many more years to follow. 1 year ago, in the first workshop, we wanted to understand and know from the PIs of the asked 6 questions, and here are the outcome or the reports. Was are you still active in the in terms of output, you can read here. You can read here what was anticipated. There is already some working groups initiated or defined. So half of them roughly. Then, how many people responded? You remember, actually, that was openly phrased, and whoever was interested could join. And here is the outcome of the responses, how many people got in touch with the PI or the group of PIs. So let's say in the majority, there were not so many people, as you can read here, but still some, which is good, of course. We also asked the question about how much time is required for completing. Overall, maybe to keep this short, the impression is that people are still committed, so no one abandoned. However, as we all know, time is limited. We have all here in this QNet is, of course, spare time or extra time, and that is probably showing in the outcome of this questionnaire. But anyway, so that was the update to the projects, and we hope, of course, to stimulate. There was another also activity meanwhile, which is maybe something you have seen. So there was an article announcing QNet. So QNet, a new scholarly network on quantitative wood anatomy. It was written by the coordination team members, but we also acknowledge, of course, everyone who contributed. You can read this in the acknowledgements. And basically, you know, this is maybe a reference you can cite whenever you think that's reasonable, and that could be then the paper for that. Good. So that was our introduction and welcome. And so you are still muted. You want to?
Can you listen to me now?
Yeah, now we hear you.
Okay. Sorry, I do have a kind of a problem with the internet connection, but it's very, very slow. So it seems that now it's working better. So the first speaker will be Peter Gessen. He's a research leader at the Royal Botanic Gardens in London, and he research mainly focused on wood and timber in the context of systematics and conservation. He's specializes in the anatomy and identification of hardwood and softwood, especially in leguminous species and Malpighiales species listed under the Convention of Trade Endangered Species. Today, he is going to talk about identifying tropical woods. Peter, the floor is yours.
Right, can you all hear me? Yes, so the next thing to do is the challenging bit of sharing the screen so that you can actually see my talk. So let's see whether this works. And I can see it on my screen. Can you see it on yours?
Yes.
Yep. Okay. So shall I get going? Get started? Okay. So when Frederick asked me to give this talk, I was slightly trepidatious because I don't do a very much quantitative wood anatomy. Most of what I do is look at patterns and identify woods using the patterns. And as you can see here, you've got quite a range of different tropical woods, including Caesalpinia echinata, Agonistylus, a Pterocarpus, a Pericopsis, and a Podocarpus. So
Peter, maybe you still want to move to the presentation mode. So
am I not in it already?
Beautiful pictures.
Oh, right,
we see your screen, but we see the preview.
Hold on. Hold on. Can you see that now? I better not hang around on this one because I've got a lot of slides to get through. So to put in context, this is our lab. A couple of my colleagues making microscope slides there using an old Reichert sliding microtome, and a few slides from our fireproof cabinets. We've got about 100,000 microscope slides, about a third to a half of which are wood slides. And you can see on each slide there in the middle picture that we have nearly always, we've got a TS, a TLS and an RLS of each piece of wood. We don't have very much replication, so doing a lot of quantitative wood anatomy on the limited samples we have is probably not particularly fruitful. And then on the right, you've got me looking down a microscope, and in the bottom, you can see me looking at the Meliaceae, the mahogany wood samples in an adjacent building called the Banks Building. So here, this is what we're particularly interested in at Kew, is stopping what's going on in places as shown on the left there, which is an illegally logged tree along the Rio Negro, northwest of Manaus. And there's a workshop on the right, the ship in the middle picture is Mesilaurus, member of and about all those collections, the N or the IAW. So Frederick asked me not to assume anything, so I think we can probably whiz across this slide. I'm going to talk woods, but what is wood? My definition is that it's the product of a lateral meristem called the vascular cambium. So all wood that we're looking at is secondary xylem, and we cut a transverse section, a tangential and a radial. So this is transverse, tangential and radial. So each section is at right angles to the next. So we're not really, at least in this talk, interested in woody tissue like palm tissue. All the woody tissue in a palm is the product of the apical meristem at the top of the plant. So it's not the product of a lateral meristem, so it's not secondary xylem. This is all primary tissue, so we're not going to consider that any further today. And then I use as my basis much of the time the two identification glossaries produced by IAWA, the hardwood and the softwood lists. And here is quite an easy summary. Hardwoods have vessels, tracheids, fibers, axial parenchyma and rays in all sorts of weird and wonderful patterns. And that's why wood anatomy is so fascinating. Softwoods I find a little duller actually to look at because they're far more uniform in appearance. So here there's just 3 pictures which show hardwoods belonging to different taxa. And you can see they've all got vessels, fibers, axial, parenchyma and rays, but in lots of different patterns. So the rather reticulate pattern that you see on the left in a member of the Leucicidaceae, which is the Brazil nut family, is very distinctive for that family. If you look closely, you see some small things which look perhaps like vessels, but are not. They're actually axial canals, and they're very restricted in the hardwoods. And on the right, that pattern is pretty well diagnostic of the family Proteaceae, where you've got large vessels that look as though they're sitting on a bed of parenchyma and very wide rays as well. So you can already see that most of my work concentrates on looking at sections under the microscope rather than solid chunks of wood. And I use the Inside Wood database a great deal. And there are other databases like the ones that Gerald Koch and his colleagues have produced as well. This is the one I kind of habitually use. And as you can see, it's quite a big database website with almost 10,000 descriptions and not far off 60,000 images. And it's got 3 main databases. One on hardwoods, modern hardwoods, another one on fossil hardwoods, because Elizabeth Wheeler is mainly a paleobotanist. And then fairly recently, we've put on softwoods as well. So it's a marvelous resource if you want to try and identify a piece of wood, looking at the anatomical characters. You can use these menus up here. Or if you want to know what a particular thing looks like, if it's in the database, you can search on genus or family. And with a bit of luck, there'll be a description or many descriptions of that genus and a lot of photos to go with them. So with the hardwood list, on the left, you can see that there are 163 anatomical characters. Now, with databases like this, if you're very green and you just go into a database like this, you can make all sorts of horrendous mistakes or not use it quite as well as one would like. So Elizabeth, Peter Bars and I wrote a paper on how we think it can be used. So if you want to look at that paper, that gives you a lot of guidance on how to go through it. So the picture on the right, the cover, straight away, someone who knows a little bit about tropical woods can see that you've got aliform and confluent parenchyma. You can see the rays are mainly 2 and 3 cells wide. That's a transverse section, but you can't see what it looks like in a tangential section. And in this case, you would see that the rays are storied in that particular thing. So if you think there's about 7,500 descriptions in the hardwood database, the best characters to choose if you're trying to identify something are the ones that are quite rare in the woody angiosperm kingdom. So here, rather a long list actually, the ones where less than 5% of the descriptions have that character are in blue, and then less than 10% in black. But if you think 10% is still 700 odd species, then, or 700 entries, that's quite a lot. So if you put them in combination, you will do a lot better with your identification. So I've got a couple of examples here where you've got included phloem, which is, if we go back a second, you'll see that vines and lianas are only 3% of the database, and cambial variants are only 1.3%. There's a lot that are missing in the database. But this was a very impressive search. I managed to get right down to species with only 3 characters. Concentric included phloem took me down to 69 matches. Adding solitary vessels, I didn't need to section this one. I just used a razor blade on the rather soft wood and could see the vessels were solitary. That took me down to 23. And then I don't usually like using geography as a main character, but that took me straight down to this one species, and I found some pictures on the internet, so I was fairly happy that that's what I had. One we get a lot of at Kew is agar wood, which is either Aquilaria or Gyronops in the family Thymelliaceae. And again, it's another one of those which has got very nice islands of phloem, but in this case, they're islands rather than concentric rings. So if you look at that character, that takes you down. I can see pictures of people there. I don't know whether you can see pictures of people or the actual information that's on this slide, but it takes you down to relatively few matches. And then if you put Ray's Uniseriate in as well, you get 45. And then if you put in minute pitting, then you get down to only 15. And we get a lot of agar wood in the lab, which has been confiscated by the UK Border Force. And then sometimes it's very easy. This particular piece of agar wood was bought in a market in China and is clearly nothing like agar wood. And usually inquirers like UK Border Force, they're only interested in whether something is what it's stated to be. And if it's not, they don't really care what it is. So clearly that is not agar wood. And then with macroscopy, there are several different ways of approaching it. Here are two guides which were written mainly by Spanish colleagues in Madrid, in English and in Spanish. Guides for people who are at ports of entry, who can take photographs of samples with a mobile telephone and then perhaps send them to an expert to find out whether it's worth pursuing the matter further. So this is a double-page spread of Sidrella, and you can see the marginal axial parenchyma there showing some kind of form of growth rings there. And then here are a few pictures that were in a paper from the journal Forests, and with a mobile phone and a nice surface, you can see the araucarioid pits in this Araucaria. You can see spiral thickenings in tracheids of Taxus. You can see here the included phloem in Aquilaria. And if you look very closely, you can even see oil cells in members of the Lauraceae. So knowing the characters well from something like Inside Wood and the Iowa lists, you can actually see many of these characters at fairly low magnification, even using lenses attached to mobile phones. We're also now beginning to use a lot the xyloscope that was developed in Madison, Wisconsin. And here, this really concentrates on transverse surfaces. So here you can see that must be an evergreen oak, and this must be a member of the Proteaceae. But it looks at images it takes images that are 7 millimeters by 7 millimeters. And here, this is one we know. This is a Shorea, and you can see a nice long line of axial canals there. And that's a relatively rare character amongst the hardwoods. If you put that character in, you only get about 84 matches, 47 of which are dipterocarps, and virtually all the rest are legumes in the Deuterioideae. But you can't get much further, really, unless you have a limited number of possibilities for your identity. And if you want to look at other characters like vestured pits, you really need to have high magnification, either light microscope pictures or preferably even SEM photos to see them. And then here is a few more pictures taken with the xyloscope. I don't know whether colour comes into it yet, but of course the purple colour of Purple Heart on the left is quite useful. You can see marginal parenchyma in the second and the fourth pictures. And you can see that there's aliform and confluent parenchyma in most of them. But again, this is concentrating mainly on solid pieces, transverse surfaces. Here's a difficult one. When I visited Thunen Institute a few years ago, Jorgo Richter nobbled me and asked me if I could tell the difference between these things. And using inside wood with sections, it's very difficult to tell them apart. I don't know how long I've got left, but I probably need to move through this quite quickly. So identification is much more difficult. If you're just presented with a piece of wood and asked what it is without any context, that's very difficult to do. But if you're asked, is it a particular thing, as UK Border Force often do, then that's, at least you've got some handle to work with. We're now getting quite a lot of plywood manufactured in China, and we're really interested in the face veneers of those, which are only a couple of 100 microns thick. Fortunately, they're rotary cut usually, so we can tell something from them. So here is a British penny, and you can see these layers of in the plywood, which are usually eucalyptus or poplar, but you can barely see the face veneers on the left and right hand side of each plywood. And then if you look at them face on, colours are generally some form of brownish or pink. But if you look carefully at this one, where I'm arrowing, these are actually radial resin canals in this Campnosperma. So that's a very good clue. And this is a much bigger, thicker piece, but this is a poplar veneer. But you can see you can really only work on the tangential sections with these face veneers. And then in the past, we've had a lot of wooden blinds, like these Indonesian blinds. They nearly always had ramming gonistylus in them. Certainly a bit easier identifying the woods than it would be putting the blind up. Not the most practical of characters. But a lot of these woods look very similar to each other, and it's only when you get them under the microscope. So with the ramming, you've got uniseriate rays, you've got winged aliform parenchyma, and the ray cells have very interestingly shaped crystals in them and very fine pitting. Whereas something like this Diara has latex tubes and much wider rays, latex tubes there. And this Anthocephalus, that's a member of the Rubiaceae, has crystal sand and quite wide rays and slightly bigger crystals. So there are a lot of characters that you can look at. You don't have to laboriously work your way through inside woods if you are familiar with some of these things. So here are the two main mahoganies, Swietenia macrophylla and Swietenia mahagoni. So mahagoni is at the top and macrophylla is at the bottom. Perhaps the marginal parenchyma is more diffuse in the Amazonian species rather than the Caribbean one. And then another thing we get a lot of, and I think Gerald, who's in the audience, sees a lot of this as well, is Dalbergia. A couple of hundred species, 250 species across the tropics. Not quite sure what that body is doing at the back there, but interesting scene that. And these are all bits of guitars. So a lot of the Dalbergias look very similar to each other. Under the microscope, virtually all of them look, the TLS, which you can see on the right, has short storied, mainly biseriate rays. And actually sometimes you can see vestured pits as well, although an SEM makes life easier for that. And then the patterns of axial parenchyma in Dalbergia can be quite difficult to describe and interpret. Probably Dalbergia retusa coccobolo is one of the easiest ones to recognize. And then we have used chemistry as well to identify Dalbergias. And this big tabletop here is one of the examples of how a lot of natural history collections have misidentified samples in them. If you look back at the entry book for when this table was purchased by Kew in the 1850s or so, there was a question mark beside the Bahia rosewood. At some stage in the history, that question mark was removed and fiction became fact. But in fact, it's a Hymenaea, it's not a Dalbergia at all. And that only came to light when Jeff Kite, one of my colleagues, said the chemical dalnigrin wasn't in the wood of this. So I looked at it anatomically, and it's not a dalbergia at all. So we're gradually working our way through scanning microscope slides at very high resolution. And I think Frederick has got some of our Diospora slides that were done with this AxioScan machine. And you can see here, we've got a wide range of different qualities and staining and mountants and so on. So we've got a rather diverse slide collection to work on. And I rather like this cartoon that we found a few years ago. Fortunately, we don't do quite as much damage as that usually if we're asked to identify a sample. And sometimes that's how I feel if I've got a rather difficult piece of wood to identify. Now, I don't know whether I'm supposed to be showing this final slide now or whether that's all going to go in at the end. Does Angela have something to say about that?
Yeah, so you can just like briefly eventually mention it, especially which could be the perspective. And then we do have still a few minutes for the question later on because we are just like quite in time.
Okay, so shall I say something very briefly about this?
Exactly, if you can just sum up a bit, that would be perfect.
Okay, well, we're now developing a program called World Forest ID at Kew, where we're trying to collect as many usefully geo-referenced and properly identified samples from commercial timbers. And the two questions that come up all the time are, what is it and where does it come from? So this is in the context of trade. And then if you look at some of the genera, I know Quercus isn't a tropical species, but there's hundreds of species. A lot of these things are very difficult to deal with. So there I've just summarized, and my perspective is one day will we actually be able to take a piece of wood from anywhere and tell you exactly what it is and exactly where it grew. That's a bit of a tall order, but you never know. One day we might be able to do it. And I think I'll probably stop there then.
Super. Thank you very much, Peter. It was amazing, super informative and instructive. And I was just amazed by the wonderful picture that you show us and the variability of the features. And then thank you for reminding us that before measuring and quantify anything, we first need to know which are the features that we want to measure. So many, many thanks. You can, if you want, stop sharing your monitor on your screen so we can start the discussion. We do have 5 minutes of discussion, and just to start with, and then eventually remember that we have half an hour after all the presentation that we can continue basically, just to not consume too much time. So anybody has a question, please, you can either write it in the chat or you can express yourself using the microphone, basically. So unmuting yourself.
I may start, I think, from Hamburg. At first, yes, many thanks for this very nice introduction and overview in wood anatomy, Peter. And I think it will give only a comment to additional research, because there's also a big progress in the identification of fiber material, tropical fiber material. Regarding this very important topic of international trading controls, timber regulation and others, we developed methods to identify timbers in fiberboards, in particle boards, or especially in pulp and paper. I think it's a very big demand for international control to fulfill all these requirements. And I think it should be very important in this context. It's also a good progress, especially in new topics in artificial intelligence with automatic image vision and in the identification of pulp and paper for tropical timber. Thank you.
Yeah, and actually fibers are much more difficult because, of course, they're all disassociated. It's difficult enough identifying a solid piece of wood, let alone macerated fiber.
Yeah, strongly depends only on one cell type of the vessel elements.
That, you'd macerated things like Gonistylus, which has vestured pits, and I think the maceration often destroys that character, which is one of the best
It's
characters.
difficult because we have similar or lookalike timber in this family of the Tumilaceae. And in this case, we work together with new methods, especially of chemotaxonomical identification. We developed a fingerprint, or we try to use the new modern genetic parts for this. But it's clear, it's not easy, because you have to consider you have so many lookalike timber, and especially if you have to decide for Kurt, you must be very clear and sure on this.
Thank you very much for your question. We have Peter in line, and then Vicky after.
Okay, thanks. You're obviously sitting on a great treasure of wood samples and also of scanned wood samples. And one question is, are you open to collaborations of now quantitative wood anatomists that would like to make use of this treasure? Mm-hmm. And as you've scanned, as you are scanning many of your slides, so this could easily, or not so easily, but in any case be used for quantitatively measuring something. And the other question in respect to that is, what kind of metadata do you have? What do you know about your samples, where they come from, which kind of forest they grow in? Because this is to some extent important, because there is a substantial intraspecific variation, which we may have to want to account for.
Yeah, I think a lot of the specimens in Xylaria are quite difficult to use by people like yourself, because quite often we don't know quite where in the tree the sample came from, what the cambial age is. You might know that if you've got the center of the stem in the sample. We might even, it might just say Brazil or Thailand or whatever. It doesn't necessarily tell you as much as you'd like to know. That's one of the things we're trying to do with our World Forest ID, is we want to know precisely where the tree grew, so properly geo-referenced. And that's mainly not from the ecological point of view, but actually trying to check things like paperwork. Like if you're saying that the face veneers of this veneer came from the Solomon Islands, and the stable isotope pattern doesn't match the Solomon Islands, then you can say, well, someone's telling porcupines with the paperwork, so we don't trust it. It's not going to be much help ecologically, I don't think. So the samples we're getting in the future, you will at least know precisely where they come from. Whether there's enough material to actually do the kind of work you want, I'm not sure. I mean, Frederick, who might be in here somewhere, we sent him some Diosporus scans of woods that we've scanned. But of course, they're just a limited area of a large part of, you know, it's not a massive cross section or anything. It's only a few millimeters across. So I don't know. What do you think? Do you think that's as useful as you'd like?
It depends. It strongly depends on the question people are asking.
Yeah.
And so I have no precise question I will ask you, but to be aware of these kind of samples that are sitting in your and in other Xylaria is a good start. And then if people are interested, they might ask you what kind of additional information there is, or which kind of additional information they might want to have.
Yeah, certainly. Well, I think our collection at Kew, as it is, the main Xylarium, and I think the ones in Leiden and possibly Hamburg as well, they kind of reflect colonial history for a start. And a lot of samples are trade samples rather than botanical ones with vouchers. Some of the more recent ones which do have vouchers are from branches rather than sort of commercial type trunk wood. So there are a lot of limitations. You know, you need to take each sample as it stands and see whether it's actually suitable for your purpose.
Thanks.
Thank you very much. We apologize for the other two questions. So please, Vicky and Reza, either write them on the chat or keep them for the question answer section later on, because we need to take track. Otherwise, we will not manage to finish all the speakers' presentation. So thank you for your comprehension. So now it's time of Marcelo Pace. He is an assistant professor at the University Universidad Nacional de Autónoma de Mexico. He's a systematic plant anatomist interested in the evolution of anatomical diversity in plants by integrating data from ontogeny and phylogeny. His research mainly focused on the evolution and the development of climbing woody vines, such as lianas, for example. And today he is going to talk about the diversity and the evolution of wood anatomy in Sapinales, problems and solution with the limitation of quantitative characters. So the floor is yours, Marcelo.
Thank you very much. Let me just see how I share here my screen. Just 1 second. Okay. Okay, I think it's working, right?
Perfectly working, nicely.
Okay, so first of all, thank you so much for having me. It is an enormous pleasure to be part of this group and to be able to present something on tropical woods and our work here at the National Autonomous University of Mexico, and of course, my collaborators. So as Angela said, I'm going to talk about a little bit about diversity and evolution of wood anatomy and Sapindalis. And there will be some, so this is a joint project. I will address this later also, but it was done here at the university in Mexico, the National Autonomous University, also with collaborators in the University of São Paulo and the Cornell University in the US. So let me just see here. Some disclosures before I start speaking about the science per se, but of course, this project could not have been done without a group of experts, because we analyzed 257 species of Sapindalis, of different families of Sapindalis. The work was triggered by two professors of the University of São Paulo, José Rubens Pirani and Diego De Marco, who are editors of the Brazilian Journal of Botany and are creating soon to come special edition of Sapindalis. And you will see that a lot of the problems that I will address during my presentation are related to the fact that we wanted to understand character evolution in Sapindalis. We had a well-supported phylogeny from Milner and Hill from 2016, but we did not have the exact same species, and we wanted them because that's the best. And so we have resourced a lot to Inside Wood that I'm happy that Peter Gasson has spoken about just now, so you're familiar with it now. And of course, our own personal collections. So, but for some families, we had amazing collections, like Teresa Terrazas is an expert in Anacardiaceae, so she had actually a lot of slides. But for other families, we do not have the same rich collection. Although we do have a wood collection in Mexico and in the National Herbarium, which is quite good, and unlike many collections, it is almost completely vouchered. So the order Sapindalis is not an unknown family for most of you. I am very sure that everybody had a lemon juice once, or ate a mango, and lychees, and the maple. And these are just some examples, but there are many, many interesting species in this order. The order Sapindalis, when we look for treatises on them, you will see that they are mostly distributed in the tropics, the bulk in number of species, but you can also see that they have occupied and diversified also in the temperate regions. I am sure that everyone is familiar with Acer, which has both important woods and are very famous, especially for Canadians, with the maple syrup. And in terms of habits, being such a large order, of course, it has all the possible habits known for plants, but most of them are trees, either large trees like this Cedrela dorata, or small trees, shrubs like the very aromatic ruta from the Mediterranean. And lianas are not so common, except for Sapindaceae, where there is a large tribe called Pauline that contains numerous lianas. I think it's over 200 species of lianas. And in Anacardiaceae, we have the poison ivy that is not very liked by people in the temperate countries, because both in Asia and Americas, because it has substances that can create a very strong allergic reaction in the skin. And if you think of mango and if you think of lime, and now I just spoke about the poison ivy, where are these substances coming from? So these plants contain numerous secretory structures. Some of them are depicted here. So for example, here in citrus, we have large cavities, also oil glands in the leaves that can give the special aroma that we like. And here you see a section of liana of Sapindaceae with an exudate, and white exudate. And when the studies were made by Israel Cunha-Neto and collaborators, they found that they come from laticifers present in the secondary phloem and in the pith. But also with most interest for our audience today, they contain also some of the most famous and prized timbers in the world. So Peter also mentioned mahogany wood. So Swietenia macrophylla, Swietenia mahogany, they are some of the more expensive woods in the market. And also here, Spanish cedar, the Cedrela dorata, we have crabwood, andiroba in Portuguese, and many other important woods. And the order is not so easy it wouldn't be possible to say one feature specifically that define the family, the order, because it's very varied. So we have from a variation like in Acer, where we almost have no parenchyma. We have vessels solitary to multiples of 2 and 3. Then we have in many groups plants with wood standing to radial chains and marginal parenchyma. And here in Sapindo saponaria, you can see the abundant aliform confluent parenchyma. So it's very varied. And we had the phylogeny. So with the phylogeny, as I mentioned, it's a well-supported phylogeny with the 9 families. We were able we first screened the entire variation of the family. We tried to delimit characters. It was easier in some cases. It's much easier with the qualitative characters. So here you can see we were able to delimit 22 characters. It is very easy when it's, for example, presence of silica, present or absent, or presence of traumatic canals. Also, either there are canals or there are not canals. But we faced a problem when we were looking at quantitative characters, because obviously they are important too. Sometimes you see, and the rays are extremely wide. We can see the number of parenchyma per strands in tangential section. And we didn't want to miss this information. But we did not have the samples because they were from inside wood, from different sources. So we couldn't measure. Therefore, we had to discretize these characters and create intervals, which is not ideal. And I will show you when you use the algorithms and the models for phylogenetic reconstruction, there are clear problems. So for example, intervessel pit size, for some samples, the samples from ourselves, we could measure, but from the collection, we could only say they were lower on or wider than 8 micrometers. Another example here, perhaps, let me see, because the arrow comes in the middle. Just 1 second. Oh, I don't know. But there are many other characters, for example, wide rays. Why less than 3 cells? Why this character and why any other one? So obviously it's not ideal. And I'll address that soon. So first I will show some qualitative features. And as I said, they are less controversial. So ring porosity in the family being mostly distributed in the tropics, we see that the ancestral state reconstructed for the order is having diffuse porous woods. But then with multiple evolutions in all the families of ring porosity or semi-ring porosity. And we found a positive correlation doing Pagel 94 test for having helical thickenings when the wood was ring porous. Something that is not new, it's well known. So basically what is happening here? Why some woods evolved ring porosity? Basically, it's also the well-known correlation with climate seasonality. I see that there are many dendrochronologists here in the audience, so they know much more about this than I do. So here, with the alternation of the very cold season and the hot season of the summer, we create the ring porous woods with very large vessels in the early wood and very tiny vessels, or sometimes even tracheids in the late wood, here in Arutaceae. But also not only the temperature is a trigger of ring porosity, but also the seasonality of the rainy season and the dry season. This is a photo of the Tehuacan, Cuicatlan region in Mexico. This is me and a colleague collecting. So you can see this is during the wet season, so it gets much drier, and all the, except the cacti, of course, but all the other plants will get brown. And the woods also respond, creating a ring porous or semi-ring porous wood. Marginal parenchyma marking growth rings. So whoever is familiar with, for example, mahogany knows that it has marginal parenchyma, right? And we found it to be very common in the order, except for some cases. Here in this Burseraceae, you see that the growth ring marker is the narrow fibers. When we reconstructed that to the phylogeny of the order, we saw that the presence of marginal parenchyma was reconstructed as likely ancestral in the order, and being largely present in many of the families, with a loss here in Burseraceae and Anacardiaceae, where you don't see so commonly marginal parenchyma anymore, and also a loss in the subfamily Sapindoide within the Sapindaceae. A storage structure is also quite important and unique character. So remember that Peter Gasson mentioned that we have an interest in more rare characters because they usually are more specific. They have a stronger phylogenetic signal. So here what we found was the storage structure is absent in the order, was in the ancestral state for the order. Probably the ancestor of all the Sapindales many years ago did not have storage structure, but it evolved in some families. And here they are especially common in these two large families. I don't know why the names were cut, but this is probably Meliaceae and I'm sorry. And here I am not sure. So Rutaceae and Meliaceae have this feature as ancestrally present for these orders. I hope I'm not speaking too fast because that's a tendency for me, but let me know, please. And here the ray composition. So the orders and Sapindales usually have heterocellular rays. Homocellular rays are more uncommon. You find in Acer in some groups, but the prevalence is for heterocellular rays. Here you can see then the red color marking that the presence of heterocellular and co-occurring with homocellular is the most common feature, with then the evolution of different types, and exclusively homocellular is present in some Rutaceae and some Sapindaceae, and in Nitrariaceae, which is a very small family. But what about the quantitative features that I have been talking that they are problematic? So indeed, our main problems were that we wanted our sample to match the exact taxa from the phylogeny that was already published. And Inside Wood does not have the exact measurements. This is one of the pleas that one of the things that I would like to ask the community, that we start presenting the exact data, because that could be used in the future. Because since the intervals are always arbitrary, I will show you the problems. And here, before I show the problems, I'll show how it should be done. This is another work that we carried in our group, but it was done with phloem. But just to show that here we had the entire collection. So for example, here we had the number of sieve areas in sieve plates of the sieve elements. And we could use a discrete, is either simple sieve plates or compound sieve plates. But look, when we finally did the exact measurement, it was much more interesting, because we could see that these that are all put as the same thing are actually quite different. So there is diversity, and you can sort the species and you can understand evolution better. Another example is also in the quantitative, the quantity of fibers. Again, if we're discretizing 3 features, we see it's very confusing. Why, when we measure exactly the quantity of fibers in these phloems, we were able to show that while in one lineage, the quantity of fibers in the phloem is diminishing, in the other is increasing. And we could correlate that with climate and the growth of these lianas. So, but when we do not have the possibility of quantifying then you have to go to intervals. I'll show, for example, here the correlation between tyloses and intervessel pit size, and especially ray vessel size. This is not something that we discovered. This has been said, even Chataway was one of the big advocates of this theory, that you only have tyloses when the pits are large enough to allow the pit membrane to expand inside of the vessels. And before Chataway, some German authors also had already seen this correlation. So the hypothesis put forward by Chataway was that exactly large pits are necessary to the entrance of parenchyma inside of the vessels, obstructing the vessels in the hardwood. And indeed, what we saw was a strong correlation of the only groups, the only families that had tyloses in the Sapindalis are Anacardiaceae and Burseraceae and Kirkiaceae, which are sister groups. And they correspond exactly with the ones with large, higher than 8 micrometers pits. So this is very interesting, of course. In terms of axial parenchyma in Sapindalis, you can see the variation that we found from woods with almost no parenchyma to woods with a lot of parenchyma. But here we did not have the possibility of measuring, as I showed you was done before in the Bignoniaceae. So basically, we could largely see only that the scanty axial parenchyma was the ancestral state with the evolution of more abundant parenchyma. But it would be ideal if we had the quantities, because probably a lot of these, because this looks quite too varied. Probably it's not what's happening. Probably it's a quantitative thing happening here behind. More problematic was this one. You can see that the algorithm got crazy. Basically, we were measuring how many cells are in each strand of axial parenchyma, and we had a lot of variation. So we had some that had only 2 parenchyma cells per strand, while in Cedrela or Mahogany, for example, you have 8. And we were trying different ways of discretizing this character. It was, in all instances, was as crazy as what you're seeing here. So basically, the algorithm, the transitions were so frequent that the program was saying, so whatever thing can happen. So here you see that the branches mean the populations. It would be so labile that it would be back and forth constantly between one feature and the other if this were true. So this is not the true history of this character. The problem is that the discretization is messing up the capacity of the model to infer an ancestral. So basically, we couldn't present this character. We just mentioned the problem, because the model is estimating a biologically unreasonable number of transitions. It's not so easy to pass from 8 to 2 cells per strand. Any wood anatomists know that it's not so labile. And here, way width, this is a little bit better, but also showing that from thin rays in blue, I'm sorry, I didn't put the legend here, it evolved many times in wider rays, but also it would be great if we could have the exact numbers of cells or numbers of or width of the rays. So here to summarize what I presented, the ring porosity is derived in Sapindalis and it's related directly to seasonal environments. The marginal parenchyma is likely ancestral also, and I did not mention, I forgot, I'm sorry, but why marginal parenchyma is present in many plants, in the growth rings of many plants. So we now know, we have studies for many decades show that you have the accumulation of starch in this marginal parenchyma that is mobilized when the plants start growing. So it's related to rapid flushes of growth. Tylose is evolved in plants with larger intervessel pits, and the other lineages would have gums inside of the vessels. The axial parenchyma seems to have increased in abundance in numerous lineages, which is also some of the theories of xylem evolution in general. Axial parenchyma strand length history couldn't be correctly reconstructed because of the problems I have addressed. And that's the problem when we lose power when you use arbitrary intervals. And the ideal approach would be to measure them. So here's some general open questions. So how can we use quantitative characters in the phylogeny? We need first to understand how they change in the ontogeny. So it is known that the vessels increase in size in width and from top to bottom of a plant. So how can I use this character if it might be variable? So, and what's the extent of phenotypic plasticity of a quantitative character? If the plant is growing in a dry environment or a wet environment, does it matter? And we know that the answer is yes. So how do we standardize the quantitative characters to be able to use it in phylogenetic reconstructions or to reconstruct characters? And how to avoid discretization of characters? I think this one is easier, but then we have the problem that we have large collections. We have the amazing database that Elizabeth Wheeler and collaborators have created. But how do we extract the data from there in a short amount of time? Because people give you a deadline. They want the paper in three months, right? So that's a problem. And since most wood anatomy descriptions standardize based on their IAWA lists, and there are many criticisms on these lists because they say that the lists have arbitrary characters, which is true, but the lists weren't created for this aim. So it's our job to adapt what is in the list to the aim of our study. We cannot just criticize the list without like understanding the lists were created to identification. And the outlook is that we have an open like avenue of new possibilities because we have very, very robust phylogenies. We have a lot of phylogenists and systematics wanting to collaborate and understand how to use their phylogenies. We have the woods, so it's time that we increase more and more the understanding of wood evolution crossing both information. And I strongly believe that like the tail studies in monophylletic groups will really increase our knowledge and allow us to address questions on evolution of wood anatomy. With that, I would like to thank you. I thank also my institution. And here's my email and website. If you have any question or would like to collaborate, I'm very open. And thank you very much again for the opportunity to talking to you.
Thank you, Marcelo. It was fantastic, and you were perfectly in time. Super. So we can now start the discussion. We do have 5 minutes, about 5 minutes, and then if we do not manage to finish the discussion, then we can continue afterwards. So you can stop sharing so that we can see everybody, and then. Super. If anybody has a question, please just write or type it into the chat or just express yourself.
See Vicky Vickers is the resident.
Yes.
Hi, Marcelo. Thank you very much for the presentation. It's really great that you incorporated analysis into a sort of an entire order. I think that's really useful information. But I was wondering about the influence of plant size. Have you looked into that? Because I was thinking about the ray composition that you would expect homocellular rays to be more common in smaller plants, right? Have you looked into that? Yeah,
so if we follow Cribbs, like the studies that were done by Cribbs and Barkhorn, we see that even a plant with a homocellular ray, in the beginning of the ontogeny has heterocellular ray. And so, yeah, but you only use adult wood. And because we had vouchers for many of the plants, we don't know the vouchers for the inside wood, but we know that the sources are usually adult plants. But yeah, it's complicated. For example, for Chondroanthus or Tabebuia, they are homocellular rays and they have homocellular rays, but they start heterocellular if you sample close to the pith. So you always have to avoid that. But when you see a section of a wood, for example, the wood behind us, you can know that it's not very young wood because when the wood is very young, the rays will be with an angle. If they are parallel, you know they're already far away from the pith. So you expect to find adult wood already. I don't know if that answers your question.
I think it does.
But in shrubs, usually the rays are heterocellular, not homocellular, on my experience at least.
And with lianas? Do you know?
With lianas, so lianas usually have the prevalence of heterocellular rays. It's very typical of lianas, even in families like Bignoniaceae, that's the family that I know better. There are many trees that have homocellular rays, and when they evolve lineages with lianas, the rays get extremely heterocellular. And then you go back to Carlquist's suggestion, because Carlquist suggested that homocellular arrays would help this distribution on radial direction. But when you have a thinner stem, it makes sense that you have heterocellulase, because then you have also some conduction on the vertical side, helped by the rays, right?
Okay, great, thanks.
Thank you.
Georg?
Right. Thank you, Marcel, for the insightful presentation. Maybe I have rather a comment than a question here. It seems to me for people like you, maybe also Peter Gasson, you would like to have as little variability in the traits or plasticity in the traits as possible within a species, right? Whereas for many of us who try to link quantitative traits to environmental variability, this is actually the variability we need to work with, right? It's more a comment that maybe came to my mind more clearly when I listened to both of you. And probably, of course, and this we will hear a bit later by Peter Hietz, of course, also the standardization that you mentioned is a very important, topic. So looking forward, and thank you very much.
Thank you.
Robert, do you have a question?
Hi, I guess you're talking to me, right?
Robert Muscarella.
Yeah, thanks. Really interesting stuff. I mean, just following up on that last comment, I wonder if you've considered using the amount of variability itself as a trait, as kind of like that perhaps different species are exhibit different levels of plasticity. I mean, that I wonder if you've thought about doing that somehow.
No, I have not. It's really interesting. But I imagine that we would need a smaller group. Probably we would need, for example, I would choose still a monophyletic group, but with less species. So we were able to have many specimens in one species to be able to actually reflect the variability in the population. Because what we had like, for example, in the best scenario, we had 5 specimens per species. And some we had only 1, so we had to rely on that number, right? Which is far from ideal. But yeah, that actually, I think your idea is great, and I don't think anyone has done that, to study in a monophyletic group and with a large sampling of specimens, how the variability is, and it would be great. Yeah, I hope you will do that.
I was going to say I can't wait to read your paper doing it.
That's great. So new ideas. Peter, please.
Yeah, it might be good at looking at something like a very fast diversifying genus like Inga, where you've got hundreds of species which are supposed to have a fairly short geological history or time frame amongst them. And I would also like to think that as wood anatomists, we can trust the taxonomists in that that a genus is a genuine, entity, and that there's a fair bit of uniformity within a genus. If the taxonomists have got it right, you shouldn't find too much qualitative variation within a genus. Like, say, Quercus, for example, you might have split it up into evergreen oaks and red and white oaks, but beyond that, the species within those groups should be pretty difficult to tell apart with the anatomy.
Mm-hmm. And I think your experience especially tells you that, right? You have seen so many samples, and I also have the same impression. I think that the large quantitative changes happen when in a genus like Dalbergia, sometimes you have the evolution of a liana inside of that group. Then you have a big switch to much larger vessels or much more parenchyma. But in general, if there are no switches in habits, I also find it to be quite homogeneous.
Yeah, I do like to try to be able to make generalizations. Like one I would like to make is that Dalbergia has biseriate storage rays and Pterocarpus has uniseriate storage rays. But then when you look at the Madagascan ones, they clearly haven't read the textbook, because a lot of the Dalbergias there have uniseriate rays. So virtually every generalization I want to make, someone's going to come up and say, well, what about this or that?
Yeah.
So
Exactly.
there's the last comment before we move to the next presenter. So it's from Peter Hietz, and it's about the last question from Robert. You would need also more specimen and know the gradient, the size, the climate across which these were sampled probably also. Okay, so if we agree, we can move on to the next presenter. She's Rosana Lopez. She's an assistant professor at the Universitat Politécnica de Madrid in Spain. Her research mainly focuses on understanding the triggers of tree mortality under drought stress, looking at mainly plant hydraulics, drought resistance, quantitative genetics, but also intra and interspecific variation and wood anatomy. Today, she will talk about plant hydraulics in tropical species, methodologies, issues, tips, and tricks. So the floor is yours, Rosana.
Thank you very much, Angela. So yes, I will change. Let's check if you see it properly. Okay.
That's perfect, Rosana.
It's perfect. Okay. So okay, I'm going to change a bit the topic, and I will talk about functioning of the xylem, and to convince you that quantitative anatomies at plant researchers working on plant hydraulics should work more together. And saying that, I have changed a bit the focus of my presentation. And although the topic will be still plant hydraulics in the tropics, I will mention some methodological issues you can find when measuring some tropical species. The frame of the presentation will be different strategies of tropical species to maintain water potential homeostasis and its relation with quantitative anatomy. I have decided this after reviewing the most recent literature and burning questions related to the field of plant hydraulics and the increasing interest in describing the response strategies of multiple functional types inhabiting tropical forest. So let's start talking about episodes of forest mortality, mainly caused by drought and warmer temperatures across the globe. This is a really recent map, it's still not published, based on the review by Allen, or after the review by Allen in 2010.
Rosana, may I interrupt you just a second? Could you move your microphone closer to your mouth?
Ah, okay.
So perfect, thank you.
Okay, sorry for that. Okay, again, so I'm showing you here a really recent map, still unpublished after the reviews of forest mortality studied by Allen and colleagues in 2010, then in 2015, Hallman in 2018. And after a group of people working in tree mortality thought it was time to include like new published episodes of mortality, so we updated the database of 2000 of Allen and Hallman and Hammond, but now we georeference the data so we can study where mortality has occurred and under which climatic conditions. Again, we found mortality all around the world, like in the last papers, including the tropics, of massive mortality events perhaps you are more familiar with, such as the one in Central Europe here, killing thousands of beech and spruce trees in 2008. In a context of warming and more frequent extreme events, the effect of future drought will almost certainly be worsened because of the increment in the temperature associated with global warming. When we plot these mortality events across biomes, we notice that most mortality events have been reported in woodlands and shrublands, which are by far the most studied vegetation. But what I want to highlight here is that mortality events have occurred in all forested biomes, as you see here, across a gradient of more than 30 degrees of temperature and 4-meter precipitation gradients. And the tropics are not an exception. For example, we have recently seen like the massive forest dieback in the Amazon at least twice during the last decade here. And I want to highlight this paper by the group of Roland and and put in like plant hydraulics like a big explanation of this mortality event. And this is, although this has been quite shocking and can be even surprising, Brendan Choate and colleagues already in 2012 have reported a global convergence in the vulnerability of forests to drought. In the graph in your right, they show that forest species in general operate with narrow hydraulic safety margins. I will go deeper in my next slides about the safety margin, the physiological meaning and some relations with good anatomical properties, but from a ecological point of view, a narrow safety margin implies that on the one hand, that the majority of species operate really close to their functional limits, probably as a result of maximizing photosynthesis and growth. And on the other hand, that most forests face long-term reductions in productivity and survival if temperature and aridity increases at the pace they are doing now, because they might have not enough time to adapt. And what is the safety margin? For some of you, perhaps they are not familiar with this. We calculate the hydraulic safety margin here as the difference between the minimum water potential, you saw here with two triangles, and the water potential and the water silent vulnerability to embolisms. So the water potential when the plant reaches like the 50% or 88% of loss of conductivity. So with respect to the maximum conductivity of the xylem. In this example, you have the hydraulic safety margin for the angiosperm species Bursera cimarumba, that is a tropical species, and like a Juniperus osteosperma, which is with really inhabiting like dried environments. And you will see like the big difference in the safety margin between a species living in a dry environment and the small safety margin in a species living in the tropics. Even this kind of a species can reach like negative safety margins, putting them in a really really dangerous strategy. I will show you now what is like a vulnerability curve, as Georg told me to start with the basics of hydraulics. And so you will remember from your physiology classes that water in the xylem is under negative pressures. This is also referred as under tension. As drought progresses, so as you go left in the graph, this tension increases. So you have like more negative water potential. And when it reaches a threshold, the water column inside the plant breaks and vessels become embolized. That is, they fill with air, and consequently the water transport is severely constrained. The easiest way to represent the loss of hydraulic conductivity as water potential decrease is this vulnerability curve. Once it's constructed, you can calculate P50 and P88 quite easily. I don't know if you see my mouse. So this is P50, this is P88. P88, so the water potential at which plants lose like 50% of conductivity, and the water potential at which plants lose like the 88% of conductivity. And in these pictures, I don't know if you see them properly. I have shown how these are microcity pictures. It's like you see like the black dots. The black dots are empty conduits, and the gray ones are conduits filled with water. Is like why like when the xylem is more embolized, you will see like much more black dots, meaning that these vessels are not conductive anymore. Okay, how can I construct like a vulnerability curve? So in the next slides, I will show you some methods to measure xylem embolisms. Cochard et al. So you have like extensive reviews in the literature. I have tried to summarize here in this slide. So these are like the more indirect invasive methods and the most common use until now to measure all kinds of species, including tropically. You basically need to dehydrate the sample and measure current conductivity in relation with the maximum conductivity of your sample. So some techniques have been developed to speed this process, like the centrifuge technique developed by Sperry, Alder, and Cochard, make like a kind of an improvement. So recently, some tropical species, like a pneumatic method has been employed, while instead of measuring water, they measure air. But what I want to is acoustic. I have put here acoustic emissions because you can hear the clicks of cavitation sometimes. But what experiments in the last 2 decades have shown is that some of these methods are really prone to artifacts and depends importantly of the vessel length of your samples. And this is particularly important. Here you have like what is happening with diffuse porous species. Most of the tropical forests are diffuse porous species. As it depends on the method, you have like different vulnerability curves, and of course only one is true. And this has been really related with the length of your vessel. So here I have put like a review by Anna Jacobsen and collaborators in 2012, and I want to highlight here that most of the tropical and subtropical species have really long vessels. So meaning that you only can use some methods to construct reliable vulnerability curves. And here in the bottom left of the page, you have like what is happening as if you go from this part we are simulating here a vulnerability. You increase the vessel length and you measure it with with centrifugation or air injection technique. Is as you increase the vessel length of your sample, your vulnerability curve changes for an S-shaped curve to the R-shaped curve. So your sample seems much more embolized than the real vulnerability curve implies. And I showed you this because the first thing you should do when you start measuring embolism is to measure the maximum vessel length of your species, because this will constrain a lot the method you can use. As if these were not enough, you can also induce embolism when you cut the sample, when you prepare the sample to measure conductivity. And here I show you like a picture of a paper of Torres Ruiz et al. In 2007, where they show that this is eucalypt, and even if you have like the same water potential, -3, here you see -3.6 megapascals. If you're measuring intact plants, so your xylem is barely embolized. But if you cut your sample under tension, this means that if you cut your sample when transpiring, you can induce like artifactual embolism. And to solve this, they made a protocol to try to relax your sample. You can check the paper, but it's like, this is a really good advice. Try to relax your sample before measuring hydraulic conductivity. And for that, especially with tropical species, you will need like really long samples. So these methods I have shown, like even like bench dehydration, if you measure with like a fluorimeter, or even if you measure with acoustic emissions, you need like quite a bunch of plant material. Finally, I want to show you here some non-invasive techniques that have been developed in the last years, which implies seeing the embolism as it occurs in the plant. Here you have like a video made by Hervé Cochard. This is a conifer species, but it is a microcomputer tomography has used. And you can usually, with good resolution, you need to go to synchrotrons or like really big facilities with not really easy access. Now we have like more plans, and finally, like the new optical technique developed by the laboratory of Tim Rodrigues is using Arduino, and small cameras seem to be like more affordable for most laboratories and is like quite portable. And now, because we are in a forum of good anatomy, I would like to show you how this embolus resistance of a species is like how vulnerable is the xylem to star cavitation has been related to good anatomy. So in general, like the water transport capacity and safe membranes. Regarding like vessel size and frequency, although water as here we can see in figure of the left here. So we have like vessel frequency of we see how it's related with resistance, that vessel frequency is linked to the embolus resistance. So even although in some cases there has not a final like a link, mechanistic link between vulnerability and diameter of our vessels, there are plausible reasons to suspect that natural selection could favor narrow drought vulnerability. Here you have another example. This is with eucalypts made in is like covering a huge climatic gradient in diameters and enrichment are quite linked to like the precipitation gradients, the eucalyptus growth, and they related to like having two strategies of eucalypts living in arid habitats having a safer xylem, while eucalypts living in more humid habitats having a more efficient xylem with much larger vessels and more frequent. And on the right you have like a study, this only with tropical species, and they show more or less the same tendency. Okay, so let's talk about now about I told you that I was going to focus my presentation in the hydraulic safety margin, and I have been talking about like the P50, so the vulnerability to embolus score. So let's talk about the other component to calculate the hydraulic safety margin. So like the an intrinsic property of your xylem. So the minimum water potential at tree reach is a function first of the environment, like recent precipitation, water holding capacity of the soil sample, and other plant traits. Such as like how the tree control water, water lost with basically by epidermal conductance of stomatal regulation, and how which water has the plant accessible, mainly with rooting depth and the capacitance of the xylem. So in a recent study, Oliveira and colleagues have identified a continuum of hydraulic strategies to deal with changing water availability in tropical species in the Amazon basin. In one extreme, here of discontinuous, fast growing and deciduous species with lower safety margins. So here, so we'll see here, when lower safety margin and a risky strategy, closing the stomata at lower water potential and shedding the leaves under unfavorable conditions. Of the we find as evergreen and slower growth trees which operates under high hydraulic safety margins due to first higher stomatal sensitivity, and also because they tend to construct, as we see, I say before, like a moderate resistant xylem, as you see here, like with the total annual precipitation, the green ones are evergreen species, like the orange ones are deciduous species. And so with less precipitations, evergreen species tend to construct first a safer xylem and also have like a deeper root system. So in deciduous species, this is not necessary because during the unfavorable period of the year, they shed the leaves, so they get rid of like the most of the transpiring tissue. Within the same community here, P50 is also a function of rooting depth, and species with deeper roots can access deeper soil water, buffering the change in water potential. So they do not need to construct a resistant to embolism xylem. The same occurs here with the crown duration. Here you have like usually evergreen species, and here you have like deciduous species. So if a species above deciduous, the safety margin is maintained by decreasing substantially the transpiring area, avoiding higher tensions in the xylem. And finally, and related again with anatomy, a higher dependence of axial parenchyma for hydraulic maintenance likely occurs in the tropic. This is like a review made by Morris et al. in 2016 and 2018. And they showed that tropical families have extensively more parenchyma than other temperate or subtropical species. And they have also a lot of axial parenchyma and paratracheal axial parenchyma. So trying to, so that this could just point out to like a function of this xylem parenchyma in like in the water conducting transport. And the amount of and distribution of this axial parenchyma were positively correlated with hydraulic capacitance. So buffering like extreme tensions in times of reduced water availability and allowing transpiration in tropical evergreen species as like the soil or the atmosphere get drier. This parenchyma could prevent embolus formation in the xylem or even facilitate embolus refilling, a quite controversial issue, but that I think like tropical species are really interesting to test, and is the like once your vessels are embolized, there is like quite a controversy in the scientific community about if they can refill or not. And the amount of ray parenchyma, the huge amount of ray parenchyma and axial parenchyma in tropical species could help like these species to put sugars and water in the vessels. And so they can be like good genera to test this hypothesis of embolus refill. This I only, but I would like to warmer at drying conditions. And what is the role of hydraulic stress controlling like this sensitivity to drought? I think this is particularly relevant because like in the tropical species are extremely diverse, we have much more data of hydraulics and also of xylem anatomy or temperate species. So my outlook, we can discuss later, is like we need to understand and predict the dynamics of tropical vegetation in scenarios of changing water availability, and to try to introduce like more hydraulics or anatomical traits in vegetation models. And that's all.
Thank you very much. Very interesting topic. We can start the discussion now. So if anybody has a burning question, please feel free to unmute your microphone and express yourself. Yes, exactly. Meanwhile, Rosa, you can, perfect. Reza, please go on.
Okay, hi, I'm Reza Ouladi from University of Tehran in Iran. I'm so happy to be here. Before asking my question, I just want to express my condolences to Professor Eckstein's colleague, friends, and family. He was a great man. That was a really interesting presentation, a very sophisticated one. Thank you for that. I just have an experience, similar experience, but of course not as sophisticated as you did. We studied the dieback of kind of Kirkes brandti Persian oak in Iran, and surprisingly, we found out that trees, oaks with bigger vessels, they are more susceptible to dieback, to death. So, although you are right that bigger vessels are not more safe, but on the other hand, maybe it shows that tree is more vigorous, more healthy, so it can react better to the changing climate or to the pathogen. So I think we have to take it into account. And I just want to ask, did you have, I mean, I think according to your presentation, your result is quite contrarily to what I say. The small trees with the smaller vessels, they could handle better the worst climate. Is that true?
So, but related, sorry again, press again. I think this is the problem with strategies. So it depends of how far you go or how stressed they are. So in general, I have to say that I have barely worked with tropical species. I have mainly worked with Mediterranean and really xeric species. So diffuse porous species is another history. And also I think with Resproters is another history too. So it's like there are many functional traits strategies. So they are many functional and functional strategies trees can follow. So that's why when I have been reviewing the literature about like tropical species, I have found, especially because of the high diversity of these areas, I have found this quite useful, like between like fast growing species with really is like that, how do you say, it's like live fast, die first or something. I cannot remember the exact sentence now. And I think in these tropical habitats, they follow quite well, like this kind of continuum. I don't know if
yeah, I get it. You mean if in different regions with different climate or different species, maybe so, different strategies.
Strategies mainly because you have to take into account like the length of the drought. That is nothing to do in tropical sites within Mediterranean, or I imagine in Iran is like I find it more similar to Mediterranean areas. So I think like the strategy is to be totally different. But the important thing is like to keep in mind is like to survive drier conditions or warmer conditions, trees have to maintain like this hydraulic safety margin. So if they cannot reach like a water potential in the xylem, then that cause like massive xylem failure. So which strategy you use, so you can follow like constructing a really resistant xylem to embolies, a really resistant xylem, or you can try to avoid reaching like these dangerous levels of water potential in your xylem. So this is like, if you have like this in mind, then you, from this point, you develop like different strategies. And of course, in different biomes, like the strategy will be different.
Thank you.
Thank you very much. The last question before the break, and it's in the chat from Fabio Genaretti, very practical one, very interesting one. So what is the more reliable and less expensive method to measure conductivity according to you?
Oh, so I think in this I have more experience than measuring tropical species, I have to say, but it's like what is the most reliable is like seeing the embolism in intact plants. But from my point of view, especially if you measure in the field, we are far from bring a portable tool to the field and see your embolism. So I think the most similar to that is like the small Arduinos and cameras developed by the laboratory of Tim Brodribb in Tasmania. But to be practical, and if you want to measure easily, I think the most practical thing is to have a flowmeter and take care while collecting your samples. So because even instead of using a flowmeter, you can use a balance and measure the water flow through your sample. So the only thing is that you have to take care of introducing artifacts in your measurement. So that's why it's like hydraulics is cheap to measure, is really portable, so you can measure in the field, even in remote locations. We have experience with that, but you need to be really careful because you're measuring a really unstable system. And then we can follow in the discussion if you want. So a flowmeter or a balance is my, and then you leave the branches to dehydrate. The problem I want to say while collecting samples, especially in tropical trees, if you're measuring like really tall trees, is that for the branch dehydration techniques and the cheaper techniques, you will need a lot of plant material, and this can be an issue sometimes.
Perfect. Thank you very much. And when I have a follow question, but I will keep it for later.
Okay, thank you.
Thank you very much to all the speakers so far. It was great. And we have now 50 minutes of break, so we can take a coffee, and then we come back 5 minutes after what we have planned, because Georg is now more flexible and less Swiss at the moment. So we see each other at 4:5. So 5 minutes past 4. Here the same. So see you later. So now it's time of Peter Yates. I hope that the pronunciation is the correct one. Otherwise you will repeat after. So he is a full professor at the Institute of Botany, the University of Natural Resources and Life Science in Vienna, Austria. His research mainly focuses on plant ecophysiology, water relation, and ecology of tropical plants and functional anatomy. Today, he's going to talk about how size matters in terms of structure and functional relationship that are linked to tree allometry. The floor is yours, Peter.
Okay, thank you. I'm sharing my screen and my title. As for my name, I'm Peter Heitz, not Heitz and not Heitz, but I'm very relaxed about your pronunciation of my name. I'm from the Institute of Botany of the University of Natural Resources and Life Sciences, BOKU in Vienna. And when Sabine and Georg asked me for a presentation, I said yes, and later they asked me for a title. And I had very little time to think about the title. And also had very little time to think about what I was talking about. And I just gave them the very short and catchy title, Size Matters, because I'm talking about size, which pretty much everyone who measures something is talking about. And this short title gives me the opportunity to talk about tree size, which is part of my talk, but would also give me the opportunity to talk about vessel size, which is also important, obviously, for physiology and for quantitative wood anatomy, which I also want to talk about. But then I didn't want to invite people to our QNet workshops who think about things other than trees and vessels when I talk about size matters. So we got the subtitle, Structure and Function Relationships are linked to tree allometry, which is part of what I want to talk about, but not only. My approach is, as was said, I'm an ecophysiologist. I work with tropical trees, tropical species, tropical forests, and I'm impressed by the huge diversity in tropical trees, and I want to understand this diversity in many aspects, including in the tremendous diversity of wood. So the question is, why is wood so diverse? So what different strategies do trees have by building such different wood structures? A little bit on my approach to quantification, which I'm a bit embarrassed to say in this circle, and particularly with Georg von Arx, who developed a very nice and sophisticated software to quantify automatically wood features. But this is not what I use. So we manually mark vessels, mark fibers or rays, and mark parenchyma. And this is the way we quantify wood, which is slower, but a safe way if you want to work with many different species. And I bet there are ways of doing this faster and adapting software to measure what we're measuring essentially by hand. But then if you work with so many species and have a few samples per species, my guess is that it would take very long to adapt this software to those various species. And this is maybe also a request for people working with sophisticated approaches or more sophisticated approaches to quantifying wood anatomy. How could we use software to extract features from many different types of wood? So what kind of wood traits I'm extracting you would be familiar with. Wood density is something also measured from the samples. Then we measure vessel area and maximum vessel area, vessel density, vessel lumen fraction, which is the proportion of the cross section that is occupied by the lumen in fractions. We calculate the hydraulic conductivity, actually, or the theoretical hydraulic conductivity based on vessel sizes and density, and to some extent also measure other cellular fractions. So that's axial parenchyma, radial parenchyma, in fiber fractions. I was told, or I understood, that the purpose of this talk is also to give some hints of introduction to people who want to do more in quantitative wood anatomy. So I'm also telling you a few very basic and rather trivial things. And one of those trivial things I want to give you is that when you're working with vessel sizes, obviously you can measure either vessel area or vessel diameter. And much of the older literature, I think, measured vessel diameter because this can be done with a ruler. Now that we use image analysis software, mostly we directly obtain the vessel area. Now, both are fine measures of vessel size, and if the vessels are spherical or round, it's easy to convert the vessel diameter to the vessel cross-sectional area. Sometimes they are not perfectly round, but let's leave this apart. But you should be aware that if you measure individual vessel diameters and then calculate the mean diameter and use the mean diameter to calculate vessel area, this gives you very different values from measuring individual vessel areas and then counting the average vessel area. Be aware that using different kind of data is tricky when you want to pool them and when you want to calculate new things from different data sets. Now, one question where this affects our data analysis is, for instance, the consideration that we have two types of wood, same cross-sectional area. The one type has 4 large vessels, the other has 16 smaller vessels. So the mean vessel area here is, we have twice the diameter here, the mean vessel size is twice as high. the fraction occupying the vessel by vessels is the same, and the hydraulic conductivity would be 4 times higher here than here. Now, do trees adjust to this by having, if the vessels are larger, they tend to be fewer, but they could be still fewer and occupy a lower proportion of their cross-sectional area if they have larger vessels. So my question is, do they actually do that?
Peter, you have unmuted yourself. Could you please unmute yourself? Thank you.
I was just trying to see my image. Anyway, so one question is, do trees of different species adjust the proportion of their cross-section they dedicate to vessel when they have larger vessels that transport more efficiently? And looking at a global data set with very nice compilation of thousands of individual data by Amy Sen, published in 2010, it appears that, so there's a linear correlation when you look at the log-log relationship, vessel density and size. And this is the maximum packing fraction. So in theory, if all the cross-section were occupied by vessels, we would have this line, and this line is approximately 10% of the fraction occupied by vessels. And there appears to be no adjustment. Then comparing this global data set with data we measured from Panamanian trees, or rainforest trees, where we measured each individual vessel, suggests otherwise. So with larger vessels and lower vessel density, a lower fraction of the cross-section appears to be occupied. Now, is this difference here because rainforest species are different, or because we use different methods? Looking at only tropical forest trees from their data set, we see that tropical trees follow their overall data set, although they should behave in the same way as our tropical trees. So the difference between this data set and our data set is very likely because they pool very many different data, including data of mean vessel areas, of mean vessel diameters, to calculate mean vessel areas and estimate hydraulic conductivity, which has its pitfalls. So consider what you're interested in, and if including or not including or missing small vessels has an importance. If you're interested in maximum vessel size, then the small vessels are not important. If you're interested in the total vessel fraction, then missing a few small vessels is also not important. And if you're interested in hydraulic conductivity, small vessels are also not important. If you're interested in mean vessel size or vessel density, then it does matter to miss or not miss small vessels. So one message is, be careful when using published or other data, particularly if you want to estimate hydraulic conductivity, and be thoughtful with any conversions between measures you might make. I'm interested also in vessel size, because vessel size is one feature that is related to potential hydraulic conductivity, which is obviously an ecologically important feature for trees and for tree functioning and for water transport. And other features of tropical trees we're interested in is why they grow so different. So why do they have different growth rates, or which traits do they need to grow fast or not so fast? And one trait that is dealt very frequently is wood density. So trees with light wood tend to have higher growth rates, which is not really surprising, because if you need to invest less in wood biomass, then you can grow more in diameter or in volume, if not in biomass. So that's not surprising. And we found now that hydraulic conductivity is as good or even a better predictor than wood density in predicting relative growth here, or also in predicting the maximum height of a tree. This is the relative growth rate of a species, and we sampled one or several samples per species and calculate typical species values. And this is the maximum height of a species. Now, if we look at the size of individual trees, so the size of the trees we sample, we see that actually the correlation between hydraulic conductivity, vessel size, and vessel density is better than the maximum tree size. So maybe we're not comparing traits with species-specific growth rates or sizes, but simply traits with the size of the trees we sample. Because if you sample trees, then you can't sample big trees from species that don't grow big, and you likely sample big trees from the species that do grow big. So there is definitely a potential bias here that you need to be aware of, and as much as possible correct if you want to know something about the species and understand species-wise correlations. So to be sure that this is not only based on bias, it's not too difficult to include the tree size of the sample trees in your analysis if you know this tree size. And by including the tree size of the sample tree as a controlling variable, we still see that diameter growth, biomass growth, not mortality, but maximum tree height is strongly related to hydraulic conductivity, which also makes ecological and functional sense. So what measure of tree do you take? And when we go to Xyleria, they will often tell you this is mature wood. So mature wood is what you characterize, and we know that juvenile wood looks different. Vessels are typically smaller and other things that change as trees grow. And if we can define a mature wood, so a tree above 20 centimeters or 10 centimeters or 10 years is a mature tree with mature wood features, then we would be on the safe side simply selecting these. In one study, we looked at radial variation across fairly large diameters of these here are tissue fractions, so that's axial parenchyma, vessels, rays, and fibers. And this is wood density and then hydraulic conductivity, checking whether these things change with size at all, and whether they get saturated and at a certain size range don't change anymore. And particularly with hydraulic conductivity, but also with wood density, in some cases it doesn't change, but conductivity nearly always continues to increase with size. And sometimes it increases faster with small trees, but it continues increasing, and there is nothing like a size threshold. Maybe for some individuals, for some traits, but no general size threshold where we could say this is mature wood and we don't really need to know the size of the tree anymore. So when you are convinced that size is an important thing, you can study the trait size correlation because you're interested in the structure of a tree and how a tree is organized and how the hydraulic or mechanical features of a tree change with size. And then you need to think, well, which measure of size actually do I take? A review by Barbara Lachenbruch also discussed the importance of cambial age. So how old was a tree when this wood was produced? And explain in which way these things should change with tree size, depending on whether the control is mainly by age or by diameter or by the position in the crown. And there are reasons for all those having effect. Cambial age, the development of the cells, hydraulic adjustment or mechanical adjustment. So what kind of measure should we take? Again, in the study of the Thai species I showed you before, these were species with annual tree ring, so we know the age of the sample and the diameter, and checked whether either age or diameter is a better predictor. In most cases, diameter is the better predictor. Age in some cases, none at all. But in a few cases, age actually, like these ones here, age actually is a better predictor than size. Talking about tropical trees, in many cases, I don't have the opportunity to determine the age if they don't have annual growth rings, with which many don't. And to be on the safe side, I would record and measure diameter in any cases. Now, for functional reasons, we know that a constraint or a limitation of transporting water is the length of the transporting pathway and not the thickness of the tree. So you might argue And there is a good argument for thinking, well, actually the length of the pathway or the height of the tree is more important than the diameter. There are not so many studies that report height and diameter. I took a sample that we had where height and diameter were known and simply checked whether mean vessel area or maximum vessel area show a higher correlation with either height or diameter. And it's no clear result. So the mean vessel diameter was strong, weakly, but stronger correlated with height. The maximum was stronger correlated with diameter. In any case, if you don't have any other data, try to obtain the diameter of your tree, either by measuring the diameter and taking the outermost sample, or by measuring the distance from the center, which you usually have when you take cores for tree rings. Which measure also relates to, or what we should expect also relates to the hydraulic architecture of the tree, which to some extent was pioneered by Leonardo da Vinci, who said, which is not completely correct, but anyway, he observed trees and said that the sum of the branch sizes is the same as the stem size or of the size of the thicker branches from which the sides branches deviate. Now, apparently he was not precise what measure of size he meant. Did he talk about the diameter or the cross-sectional area? Understanding trees in a bit more detail, Shinosaki and colleagues developed what is known as the pipe model theory. This essentially says each leaf needs a certain pipe or a certain transport cross-sectional area to be supplied with water, which however completely ignores the path length. And when you relate measures to the height, you see that many measures, including vessel size, but also fine scale details like the pit membrane structure, change with height. So looking at vessels only, this is not the only way by which trees can adjust their hydraulic architecture. They can adjust by modifying the sapwood leaf area ratio, the vessel sizes, the vessel densities, pit numbers, pit structure. They can increase or reduce transpiration and other things. And if you're interested in tree hydraulic architecture, looking at vessel size is certainly not the whole picture that you might be interested in. Okay. One question that we were asking is if vessel or wood features are adaptations to drought. And there are many reasons to think this might be the case, and Rosa Anna in her previous talk explained some of these. Among others, we see the correlations with P50 values, with cavitation resistance, and we see that trees in dry forest and trees in wet forest have statistically significant differences in wood structure. Like dry forest species tend to have smaller vessels compared to moist forest species, but then trees in dry forests tend to be smaller than trees in moist forests, and perhaps we're comparing small trees with large trees rather than trees in dry from trees in moist forest. So there are many biases that you can introduce into your research questions by ignoring tree size when you're looking at quantitative wood anatomy. And to tackle this, I'm working on a set of data that compares trees from different locations. So locations differ in precipitation, in rainfall, in aridity, and compares pairs of congeners, so same species, different species from the same genus, related, where one species is typically grown in wet and the other typically grown in dry species, and these are grown at the same site, or species where the dry forest species is collected from a dry forest and the wet forest species is collected from a wet forest. So that we see, this is the third actually, possible genetic and plastic adaptations, or the same species growing in dry and wet forests, in which case the differences might be plastic. So are those adaptations to aridity, to rainfall or other measures of aridity, either genetic or plastic within the species? We measured a range of wood traits, and this is where how we obtain the typical aridity or the typical climate tree species grows in by looking at the species distribution, obtaining the average annual rainfall and other measures of aridity for those species These are all the different species. So these are rather dry forest species. These are rather wet forest species. And now we'll look at the traits of all those species when they are growing in the same forest. This is the aridity or the rainfall in the driest month of more than 100 species growing at the same site. And you see very, very little, but some significant correlations. But then those trees were of very different sizes. So these traits here correlate with rainfall. If we ignore tree size, fortunately, when we account for tree size, we have at least as many or even more significant correlations. And then these tree species compositions are also different. When we account for phylogeny, we also have more or less the same significant correlations. So we identify traits that are not very strongly, but still significantly related to potential adaptations to aridity, accounting for tree size and accounting for species composition. And this is the pairwise comparisons, where I just want to show you the strength of the various factors affecting quantitative wood anatomy, be it the wall thickness or hydraulic conductivity, vessel fraction, vessel size, vessel density, wood density, and some others, where in all cases, the species or the genus identity has by far the largest effect. Wood size or tree size has mostly the second most important effect, so strong effect. And then in some cases, the question whether the trees were from or growing in dry or wet forest also has a significant effect, mostly smaller than wood size, in some cases like the vessel fraction, the vessel density not. Well, there's actually the vessel size variation, that's the Gini coefficient. This might be more important than wood size. In all other questions, wood size is a stronger factor than potential adaptations, either by genetic effects or by environmental plasticity on wood traits. And different wood traits differ in the degree to which they are controlled by either genetic adaptations, plastic adaptations, or phylogeny. Now, this is my last take-home message. You may conclude from my previous slides and also from other publications that actually the predictive power of wood traits is pretty poor. This is a publication of many P50 values, so xylem vulnerability measurements. And when you record the previous talk, there are many potential pitfalls and errors you can make, and probably there are quite a few errors in this publication as well. But anyway, we see some kind of correlation, but we see in these dry forests here, many trees with very high resistance in xylem, but also many trees that apparently are growing happily in dry forests without any of those adaptations. So you might conclude that these are not important, or that these are not the adaptations that tree have. And yes, they have other ways of dealing with drought and dealing with aridity. And this is one study similar to the study also Rosanna was showing us before, that looked at trait coordination, which looks at traits that are important for hydraulic safety and traits that are important to avoid very low water potential. And hydraulic safety often goes together with high wood density, and in those forests with evergreenness, so these are resource resistance, but there are completely different strategies of trees that have low wood density, that store water, that drop their leaves, and that by a different combination of traits deal with the same environmental constraints. So we should look at more than wood traits, and we should certainly look at more than only wood traits and include leaf traits, phenology, and others, if the purpose is to understand adaptations of trees or of plants or of organisms in generally to their environment and which traits are useful for this. So these are my summary questions, suggestions or requests for you. Report the tree size, please report the tree size, whatever your questions are, and also report the tree size so that other people may make use of your data. Which research gaps, which measure of size are relevant, are important if you're interested in tree hydraulics or this structure and strategies of individual trees. And as for quantitative wood anatomy, I focused on vessels. Many people focus on vessels because they're so easy to measure. We need more data on other wood traits. The outlook, so some of the wishes for the quantitative wood anatomy community is there are many data around. We could pool them. We could produce the metadata. Okay, there's some warning about Zoom. I'm not sure what happens, but I'm not interrupting now. And share analysis and data. And my perspectives are use quantitative wood anatomy and other traits to understand whole trees. So how they are dealing with growth, with aridity, and adjust their allometry, ecological perspectives to understand the importance of wood traits for trees in their environment, and then diversity perspectives. Why are there so many trees using different strategies, different wood structures, and why does tropical wood look so different? And my take-home message is look at tropical wood, enjoy your nice samples, and thanks for your attention.
Thank you very much, Peter. Very interesting presentation, actually. Super cool. So we do have just few minutes for some burning questions, and then we can quickly move to the last one. So please just ask either on a chat or directly. Reza, please go on. You should unmute yourself, otherwise we cannot listen to you. Sorry.
That was a really interesting topic and really interesting data. Thank you for sharing that. I really enjoyed that. I didn't want to ask a question. I saw nobody asked, so I just came to ask a question. About variation from piece to bark, you showed some data, some graphs, and most of them were linear. I was somehow surprised because usually variation of trait from piece to bark doesn't have a linear shape. It is some kind of exponential. So is that because you mixed the data of a lot of trees, and did you do it on individual tree and found a different shape? I mean, the different algorithm?
No tricking with algorithm. I guess you refer to this graph here. You see the graph now?
Yes.
So these are the fractions, and we simply used linear lines. That said, you can see that some of those trends are nonlinear. And you can try to adjust nonlinear functions, of course, and in some cases these will fit better than others. But I didn't think it makes sense to try to adjust different functions to each kind of data set. And the data are as they are, so they are not tweaked. They're generally pooled a few individuals per species, and in some cases you have faster changes initially and later no changes. In other cases you have continuous changes which may or may not be linear.
I mean, I get your point, but my question is, usually they said that by having just like a graph like that, we can distinguish or find a zone of transition from juvenile to mature wood. But here I don't see any place that we can
As
say.
I said, I wouldn't say that you can find one line that is a transition between juvenile and mature wood. It mostly changes faster initially and slower later, but you can see continuous changes in trees that are very large. Okay. Depending on your constraints, maybe mechanical constraints, maybe hydraulic constraints, and then trees continue to have to adjust some of their wood functions and others not.
Thank you. That was a great work. I should go and take a look at your publication. That was great. Thank you again.
Anytime.
Thank you very much. So if Ravo agrees, we can take your questions for later. So we can just move to the last presenters. That is Achim Broening. I hope that this time I make it. He is a full professor at the Institute of Geography in Erlangen in Germany. This research mainly focused on the effect of climate change on the ecosystem, in particular on forest, and specifically on the effect of the extreme climatic events on the functional and structural traits of organisms and their distribution, but also the impact on carbon budget. So, Achim, if you're ready, you can start.
Thanks a lot, Angela, for giving me the floor. Before I start, I want to acknowledge my co-workers and people who I'm collaborating with, because what I'm showing you can certainly not be accomplished by one person. Before I am diving into the topic, let me take a few words to frame a little bit the tropics, if we speak about them, because there are different definitions, and in the context of such a talk, it may be worth to have a brief look at different concepts, how to frame the tropics. The most common or easiest, most straightforward one is to say the tropics are the area between the Tropic of Cancer and Tropic of Capricorn. So there's an astronomical definition. However, there are lots of different climatic classifications that use different concepts to outline the tropics. For example, when the daily temperature amplitude exceeds the annual temperature amplitude, a certain span of the day length, or a lot of effective climate classification types. The most common one is still the Köppmann-Geiger classification, who outline tropical climates with a mean annual temperature of 18 degrees. For whatever reason, it is 18 and not 17. I don't know at the moment. So if we look into the diversity of tropical climates, we have, of course, not so much temperature difference. However, of course, temperature changes with altitude, but it's much more diversity in the hydroclimate. There are tropical climates with one rainy season, which can vary in the amount of precipitation, of course, but especially in the length of the dry season. There are tropical climates, if you come closer to the equator, that have multiple rainy and dry seasons. There are tropical climates where trees grow. Of course, there are hyper-arid climates without tree growth, but they are not our topic. And there are perhumid tropical climates. During my talk, I want to give some examples from each of these different hydroclimatic zones. Beside the climatic variability, tropical areas are also characterized by extremely high diversity in species, so in alpha diversity, but also, as we have already heard, the diversity in phylogeny, in the diversity of plant families and their structures, and also of tree life forms, which have certain adaptations also in terms of mechanics. And of course, as we have already heard, like examples in lianas, or for example here, the Bombaceae family with succulent trees, big variability in the hydraulic system. Most of us are probably working, when we talk about tree ring anatomy, with trees that have more or less straight stem forms. And during my talk, I will, of course, focus mostly on those examples, giving you case study examples from Ecuador, from Western Africa, and from Southeast Asia, mainly here from Bangladesh. Thanks to Marcelo's talk, I can go here very briefly about these things. There are, of course, qualitative wood anatomical features that help us a lot to distinguish tree species which form annual tree ring boundaries. Not all of them are annual, but at least they are anatomically clearly distinguishable, like, for example, the marginal parenchyma bands, or distinct fiber zones, changes in the porosity, distended rays, and others. So I don't have to address them also because they can hardly be quantified. But what is important to be quantified, at least potentially, are these anatomical features or wood features marked in red. I don't go through all the list, but I will give you some examples during the talk. What is maybe the meaning of the ecological meaning and the interrelationships of some of these variables. As we have said, some of these qualitative anatomical features like change in porosity, marginal parenchyma, flattened fiber cells are used to delineate ring boundaries. Why are they important in our context? Because they give us a dating control. So I think I will be the only speaker this afternoon who is also talking about time series of anatomical features. And it makes sense if you are working in a tropical area with a high, let's call it xylodiversity, with a high diversity of anatomical structures, to first separate those species that have fairly distinct boundaries, or that have from those who have indistinct boundaries. And if I have still a little bit time left at the end of my talk, I will very briefly address what to do with what can be done maybe in species that have no clear anatomical boundaries. One aspect that has already been mentioned in the previous talks is that the distribution of an area of a tree species, may cover a bigger area than is outlined by one characteristic climatic zone. So some tree species have a very wide geographical distribution, are quite, I would say, quite insensitive. Some of them, if they have enough water, they don't care if it's distributed over the whole year or if it's distributed to a shorter season. However, there are changes in the tree ring structure. And that is, of course, one current shortcoming of some of the available databases. If you have maybe only one description of a species that has been collected in a ever humid, in a per humid climate, it does not the same species occurs or is even planted in a zone that has a certain annual climatic pattern. This is the example of Sclerocarya from Western Africa and Eastern Africa. And you see that, for example, if multiple rainy seasons occur, the species can form density fluctuation. Another famous and very economically very important species is Tectona grandis, that originally comes from Southeast Asia, and it forms a very clear wood anatomical structure with a semi-ring porous vessel distribution in a seasonal climate with one clear rainy season and a dry season that is at least 4 months long. However, interannual xylem growth zones, which is a kind of a synonym to density variations, can occur if there are several dry seasons which are shorter than 3 months, like, for example, in plantations in Western Africa. That's just one example, but there are many plantation tree species or agroforest tree species that show such a change in plasticity in their anatomical structure. Now let's come to the case studies. The first one actually takes you to Western Africa, the so-called African oak, Afzelia africana, growing in 2 different climatic zones in the West African state of Burkina Faso. One is so-called Sudano-Sahelian zone, which has a relatively short rainy season of approximately 800 millimeter annual rainfall, and the Sudanian zone, which has a shorter dry season and a moister climate. It's closer to the equator. So we studied the anatomy of Afzelia in these 2 climatic zones. You see the species is diffuse porous, showing sometimes vessel groupings in radial arrangements of 2 to 3 vessels, and it's surrounded by aliform parenchyma. Then we separated annual tree rings by these marginal parenchyma bands, and we quantified the vessel features, in this case of the species, and compared them for these 2 climate zones. And it's obvious that some of the vessel features show significant differences, which points to the fact that tree species are able to modify, to adapt within a certain range of plasticity their anatomical structure, which has been outlined by other speakers before. If we conduct a principal component analysis on the different anatomical traits, we clearly see that some of the features are interrelated and not independent from each other. It's quite obvious that total vessel area and wood density are, for example, negatively loaded on the same factor, or that total vessel area and vessel density are, vessel number, vessel density are having an anti-correlation. So these, some of these anatomical traits are not independent from each other. Let's move to another climate zone. In this case, it's Southern Asia, Bangladesh, with a monsoonal climate, with a rainy season between, let's say, depending on the year, March or April to end of October. The country shows an increasing trend in temperatures over the last 60 years, but a relatively strong variability in annual rainfall without any clear trend. We studied 3 different tree species: a ring porous, a semi-ring porous, and a diffuse porous species. And I was intrigued to see some of the same names on one of the slides of Peter's study. Chucrasia and Tuna, Esmeliaceae species, are widely distributed. Of course, Lagostrymia is a species growing in the riverine forests. So again, we quantified many of the vessel features. You are now already familiar with most of the abbreviations. The letter I at the end here points that we indexed the area, so we removed age-related trends in the raw data. So we get here a time series of different wood anatomical features. And again, the PCA clearly indicates that some of these features show a high degree of covariation. They are not correlated with stable isotopes, but definitely vessel density shows a strongly opposing trend to most of the vessel size related features. Let's come to the climatological interpretation of such measurements. One interesting thing we found is that xylem can be adjusted in Chucrasia, the diffuse porous species, for example, in specific drought years like the year 1999 or 2006, which show significantly longer dry seasons than the average climate conditions. The anatomical features in Chucrasia show significant deviations from the long-term mean. Ring width was approximately half than normal, but also the number of vessels and the total vessel area strongly decreased, while there was a small increase in vessel density. It has already been mentioned that there may be adaptive value in such, let's say, anatomical traits. Hydraulic conductivity is certainly a very important parameter, but also the xylem vulnerability to cavidation, which is an index that you can calculate from the different vessel parameters. So we compared the 3 species, the relationship on the one hand between hydraulic diameter of vessels and vessel density, and it's, as Peter has already showed, and also Rosa Anna has mentioned, there is a negative relationship between these features, deciding a little bit the strategy if a species is more going via vascular efficiency or vascular safety. And indeed, the 3 study species, they show significant differences in their hydraulic conductivity, the hydraulic vessel diameters, and their xylem vulnerability. Well, is this important? I think it has relevance. If we look to the scientific community that is using such data, there are a lot of modelers that are working with vegetation dynamic models, or so-called landscape models of the newer generation, that are using such traits, including also P50 or P88 values, that they use it as an indicator of the potential mortality risk of a species if climate is changing into a certain direction. But not only drought can be indicated by the quantitative wood anatomical features. These here are flood years, which showed very high annual precipitation, but especially a very short dry season that is combined with flooding, with large-scale flooding. And this tree species Lagerströmia, which is usually semi-ring porous, strongly responds on the one hand with the total ring width, but also with a wood anatomical structure. So we did a superposed epoch analysis of different vessel features, tree ring width, number of vessels, vessel density, total vessel area, and also of oxygen isotopes, which are usually a very good indicator in tropical areas about the response of tree ecophysiology to hydroclimate variability. In this case, it was interesting to note that the vessel traits responded much more sensitive compared to, for example, delta 18O variability. So quantitative wood anatomical features can be a substitute, or even in some cases, maybe a better environmental indicator for some environmental events, that it is a stable isotope or other features. And this correlation pattern shows you that the relationship between vessel density in Lagerströmia was highly correlated to the mean water level of the main tributary in this study area in Bangladesh. My last example is from Ecuador, a totally different topic. In this national park in the Andes of southern Ecuador, in a perhumid mountain rainforest with a very high species richness, more than 300 different tree species per square kilometer, approximately 2,000 millimeters of annual rainfall. There was a nutrient manipulation experiment that tested the hypothesis if the performance of trees in such an environment is limited by the nutrient availability, nitrogen and phosphorus, or a combination of both. So we studied the wood anatomical features of Eichhornia lochaensis, for example. It's also a diffuse porous species which shows vessel groupings. And that was one of the parameters we tried to quantify. So the vessel grouping index is the number of vessels per the number of groupings, vessel solitary index, number of solitary index, or the ratio of solitary index, solitary vessels to all vessels, and the vessel multiple index, multiple vessels via multiple groupings. You don't have to look at all figures in this table, but here's the control group, the nitrogen fertilization and the combined fertilization of nitrogen and phosphorus. And indeed, we found a significant change in the vulnerability index. And the same, we found a strong indication in the anatomical structure regarding the vessels, and especially the multiple vessels, which is also shown in this graph. So the three colors represent the different treatments, control, nitrogen, and nitrogen plus phosphorus, and they are separated in different vessel groups per frequency, the solitary vessels, the multiple vessels, and all vessels together. And what you see is a shift. So the gray bars are longer in the left parts of the graph. That means here the smaller vessels are more represented if it's multiple or solitary in the control group, whereas fertilization in general leads to a shift in vessel size. So the species produces more larger vessels. And also it increases the grouping index of vessels. Probably the species responds via the leaves. Of course, the leaves are changing their structure, and this probably has a consequence then in the wood anatomy. A side effect may be an increase in the cavitation under drought stress conditions. The last example very briefly shows you an example that quantitative wood anatomy can still be used to some degree in species that do not have any anatomically distinct rings. This is Heliocarpus americanus from the same Ecuadorian rainforest. And again, we analyzed vessel groupings. Since there are no clear anatomical structures, we just analyzed the wood in equidistant slices of 2 millimeter width from the inside of the tree close to the pith to the outside. And we found a significant difference. The trees are all of the same size group. These are pioneer trees, not very long-lived. That there is a significant change from inside to outside in the vessel solitary index, the multiple index, and the vessel grouping index. That not so much, but in the specific conductive area. Let me conclude, and I have structured my conclusions according to my topic in the relevance of quantitative wood anatomy and in the opportunities. So first, the relevance. Quantitative wood anatomy offers strong linkages between tree ecology and environment, much more than the tree ring width can do, because tree ring width is a highly integrative parameter that is just a general indicator of the vitality of a tree. So tree ring width is limited in its interpretability and often just used in terms of statistics with environmental parameters, where the quantitative wood anatomy can provide causal links between ecology and environment, and therefore it provides very valuable additional information, especially on the effects of extreme events. In the tropics, quantitative wood anatomy is even more useful and important, I would say, than in the seasonal climates, but this remains a matter of debate. Some opportunities, adaptations in trees to the environment, may be rather seen in the modifications of anatomical structures than in the growth rate. And another important aspect is that there's also a legacy in the wood that takes some years, remains in the functionality of trees for years. Okay, another opportunity is that we can go with quantitative wood anatomy to intra-annual resolution, which is of course a new opportunity for high resolution analysis. And other aspects have already been mentioned by other speakers. The diversity of anatomical structures has to be linked to the spatial and temporal variability, also in the wood anatomical descriptions. Okay, if you want to quote something from this talk, tree ring anatomy does not mean everything for tropical dendroecology, but without it, tropical dendroecology means almost nothing. That is my take-home message for you. These are the references I used. I thank you very much for your kind attention. And these are my open questions for the discussion. How variable are wood? Well, I think I don't have to read all these things. Let me focus on the outlook. I think with the technical development of artificial intelligence and improving microscopic technologies, quantitative wood anatomy in a few years will probably become a standard parameter for tree ring analysis, like it is tree ring width or density in the meantime, and it will probably dramatically shorten the manual time that is still needed to do the analysis. That is my optimistic outlook. So thanks a lot for your attention, and I give back to Angela.
Thank you, Achim. It was very, very detailed and precise presentation. So thank you very much. And thank you very much also for the last 2 points that you raised.
I apologize that I couldn't read your message
no problem
in the
at
chat
all.
because in that view of the screen, it's
no
not
problem
possible.
at all. So we can have 5 minutes for some specific questions directly addressed to Achim, and then we can open the discussion with all the other speakers together. So please feel free to unmute yourself. I already see a hand, so Robert Muscarella, please go on.
Hey, yeah, super interesting, and I have probably a bunch of questions, but I'll just stick to one for now. Especially with the work in Bangladesh, it looked like it seemed like you have a pronounced dry season, and so that's really what's kind of your marker for separating each year. But does it rely on an assumption that you have just one dry season per year and it's intense enough every year that it causes that ring? Or, I mean, I guess I'm just wondering if for places where you might have like not such a strong dry season or potentially 2 dry seasons in a year, what can we do to try and time calibrate it?
Thanks for that's the crucial question in tropical dendroecology, actually. So the case study in Bangladesh is clearly in a monsoonal climate. The rainy season is stick to the monsoon phenomenon, and there's one dry season with cambial dormancy and one wet season. So from a climatological viewpoint, that's rather clear, rather simple case. It becomes much more complicated, for example, in the Sahel zone of Africa, where there's a much higher variability in the occurrence of, let's say, of the onset of the rains that can be delayed by 1 or 2 months. So that means the dry season can last 6 months or 4 months. And in between the rainy season, there can be a dry season or not. We have this case, for example, studying trees in Ethiopia. And with climate change, also these precipitation regimes are not stable. They are exactly the thing that is affected by climate change. So that makes it very complicated. And indeed, in many cases, we have to verify if the number of rings that we count is really correlating to the number of solar years. So we use high resolution radiocarbon analysis in some cases to confirm if the number of rings is identical to the number of years.
Thanks a lot.
Thank you very much. I may ask you all if we could switch on the camera while we are discussing, because it seems more real than instead of just having a black background. So please go on. Reza, if you want to express yourself, your question.
Okay. Hello, Achim.
Reza.
Nice to see you. I didn't want to be the only person who asked, so I just typed the question, but now I ask it. That was a fascinating presentation. Thank you, like always. About the parameters you measured, you said vessel number and vessel frequency or vessel density. I mean,
yeah, it's actually simple. Maybe it was not precise enough. Vessel number is just vessel number per rings, per whole ring, whereas vessel frequency is in millimeter square, for example.
So the first one is more related to tree ring widths. I mean, if you have wider rings.
And of course, it only works in diffuse porous species. It doesn't make sense in ring porous ones.
Okay, thank you very much.
Thank you. Are there any other questions directly addressed to Achim? Peter, do you have one?
Well, if there is still time.
Yeah, just few
Okay.
minutes more. Then
Quick question to
the
Achim.
planning.
Achim, you were talking about the vulnerability index or something like this. I guess this is the one designed, devised by Carlquist, which is some relation between vessel density and size or something like this. Is this what you're meaning?
Yeah.
Okay. I'm not quite sure how useful this is. So there are a number of wood traits that people correlate with P50 values, and I haven't seen any study that used this Carlquist vulnerability index to see how really that is related to xylem vulnerability. That shouldn't be too difficult, but Carlquist he made an argument, but his index isn't really based on any studies that tells us that trees with higher Carlquist vulnerability index really are more drought resistant.
Thanks, Peter, for this remark. I think it points us to a very general issue that, of course, now quantitative measurement of microscopic images, like we have some in the background, they produce multiple numbers, and you can create hundreds of indexes by calculating ratios or whatever between different of these parameters. And I think one of the really challenges, or one of the tasks of our community, and I actually formulated it in one of my open questions, is that we have to figure out which of these hundreds of indices are really relevant, and maybe it's also time to think of a kind of standardization, which ones we use. Of course, when you write a paper, sometimes you use that give you the highest explained variance with any kind of target parameter that you try to explain, like a standardized precipitation index or any drought index or whatever. But indeed, I think it's very important that our community thinks about which of these hundreds of parameters are really worth to not only to measure, but also to calculate, and which of them are just playing with numbers, or which can really be useful ecological indicators, in the sense that we have a mechanistic model explaining their functionality. And I really like your remark in your talk that we should not try to explain every kind of adaptation or effect of climate change just by anatomical structures, because trees don't only have wood and not only stems or branches, they have leaves, they have roots, and they have multiple ways to adapt to climate change, not only in the anatomy, also in the morphology, in the leaf area index, and in changing their porosity of the leaves, and blah, blah, blah. So we should not expect that the plasticity in wood is giving a full explanation of total tree variability. But in some cases, at least it can help maybe to give some constraints about the adaptive potential of trees, let's say like that, or to frame a certain kind of environmental envelope in which trees are able to adapt.
Thanks.
Thank you very much. Georg?
Right, Achim, I also have a specific question to you. I mean, it was about trade coordination, what you just mentioned, which was also very nicely explained by Rosanna earlier on. Now, I was just curious, in your Lagerstömia example and the monsoon and the river stream flow, where you showed that there is a close link between cell size or vessel size and, well, the amount of rain input. Was this then a shallow-rooted tree, so which makes it a more suitable hydrological meter, or can you say something about this species?
Yeah, that's a good point. I did not collect the species, but as far as I know from it, it is a shallow-rooted tree, because the point is that in the floodplain itself, there is stagnant water in the underground, which is creating an oxygen zone in the root, where the root cannot penetrate, the oxygen-free zone with stagnant water in the soil. So the trees form a shallow root system indeed, yes.
Right. So in this sense, you confirm my hypothesis, and maybe this is also now the way to lead to the more general discussion. I mean, very rarely one single trait makes a full story in terms of understanding tree responses, and probably when we have a certain target, like now in your case, river stream flow or monsoon rain input, we have to be very careful in thinking, okay, which tree species, which anatomical trait should we use if you want to simplify that much, right? Okay, so thank you very much, Achim.
Yeah, Georg, it's a very good remark. And of course, we have also to be aware that each of our models, we say they are statistically significant and they explain a certain amount of the variability of the environmental factor. Sometimes it's 50% and then we are happy. Sometimes it's only 30%. It's still statistically significant. But 30% of explained variance also means 70% of unexplained variance, and we should not forget this as
well. Okay, thank you. Can you hear me? I just go through the chat. There were some other questions, so I try to remember to whom they were addressed, but please correct me if you meant something else. I see a question from Geova Laurenco Jr. Do you think the study of the functional adaptive role of the wood traits may improve these evolutionary models? That means what trait use? If I remember well, this was addressed to Marcelo.
Yes, I think so too.
Yeah.
And I read the question before and I thought about it, and I don't think so, because I think when the features used are not related to behavior or function, well, we can be, but I don't think it's the case. Behavior is easier, but like a function, I'm not sure. But yeah, I would have to think of examples. But you need to understand clearly the function, and not everything is understood, right? Yeah, but I think the quickest answer would be no. I don't think so.
Okay, thank you. I don't know whether Geova was still here. I cannot see all the names here, but thanks a lot. And then we had some other questions were already answered. I have this one from Ravo from Madagascar. How to calculate and analyze the vessel diameter if species have ring porous and diffuse porous in the same genera? Ravo, are you still there? I cannot see. This one was addressed to Hakim, if I remember well.
Okay, difficult job for me because I think the question came up before my talk. I had also read it, and I thought it was related to Rosa Anna, but I'm not sure. So it's really difficult. That means the genus can form very different anatomical structures, diffuse porous or ring porous, depending on the environmental condition. I don't have really a solution for that, actually, but I hope that only a very small minority of species does show such extreme behavior.
Maybe comment from my side. Well, generally, if you're interested in mean vessel size, you don't need to bother whether they're ring porous or diffuse porous. You may want to use vessel size or vessel size distribution to quantify how ring or diffuse porous the species is. And then if you measure all the vessels across several rings, you can look at the vessel size distribution, or you can look at the size distribution, what the dendroanatomists are doing across the year rings, and then see how much the size, the mean size of the size distribution changes across the rings. If that's helpful for the question, but I'm not sure whether but I was still around.
Can I pipe up there? Because you're very lucky working in the tropics, because a very tiny proportion of tropical trees are ring porous or semi-ring porous. There are far more ring porous species in temperate regions.
I also would like to add to this question or remark. Well, from what you presented us, Peter Hitz, now I pronounce correctly, right? You showed very nicely that taking the mean cell size or vessel size really doesn't make sense functionally. It may be good to describe a species, but not in terms of functionality. And probably what you are then suggesting is, and there also it doesn't matter whether ring porous or not, to calculate the mean hydraulic diameter, right? If you do this, you account for different distributions of cell sizes, and you express in one single value, still in a mean value, you express the functionality in terms of water transport.
I was just reading the question again in the chat, and actually the question refers to if species have ring porous and diffuse porous in the same genera. So it's not referring to one species showing a change in the anatomy, but different species within one genus. That's how I interpret the question. And then indeed what Peter said, from a quantitative wood anatomy standpoint, it doesn't make so much trouble if it's homogeneous within the species. It's then more probably a question to the evolutionary development of such genera, as I understand the question.
Thank you, Achim. There's another question from Reza to Peter.
Yeah, hi again. I don't know if Peter is still with us or not. I just wanted to ask this question. There was no time, so I take advantage of this final discussion to ask this question. My question is about wood identification. There are people coming to me as a wood anatomist to identify soil. Maybe Professor Koch can also help me with this question, which I saw his name among participants. My answer is that the question is whether we can identify a timber just by taking a look at its features, the color, general appearance. And my answer is for temperate trees, temperate wood, there are few species that we can do that. For example, beech has some reflex that are very distinguishable, and without even taking a look at end grain, we can say that it's beech. But for tropical trees, it is almost impossible. There is almost no tree which have a very distinguished macroscopic characteristic. I mean, not end grain, but just by taking a look at the general feature, the color, and something like that, to say that this is, for example, ebony or something else. I just want to say if I'm right or if I'm wrong.
Well, if you, how long have you got? Because Gerald and I could keep you here for hours explaining what we can and can't do. But one of the biggest problems is, what do you know about the background of a particular piece of wood? For example, I think Quercus is a very good example. There's about 530 species across temperate regions. You can separate them into white oaks or red oaks or live oaks. But with the white oaks, then you might have a problem, because furniture and flooring that are made from white oaks, often in China and Vietnam, are made from North American, European and Russian Far Eastern white oak, which all looks pretty much the same. So it's very difficult to tell which species. You can easily say, yes, that's a white oak, but which one is a much bigger problem. And that's why with our World Forest ID, we're trying to combine techniques, not just anatomy, but chemistry, including stable isotope ratios, where the pattern of stable isotopes in the Russian Far East should be quite different from that in Europe or North America. So you combine techniques and work out what you've got. It often depends on what assumptions you can make. If you know you've got a piece of ash from Britain and it's from an archaeological site, then it's going to be Fraxinus excelsior. If it's a piece of ash in furniture, it could be an American one or a European one or an Asian one. So it depends on the assumptions you can make. I don't know whether Gerald has got anything he'd like to add to that. Has he gone? He might have gone.
So if I get it correct, you say that the answer to this question, that can we identify tropical trees? I mean, your example was more about temperate trees, but...
Okay, all right then. It doesn't really matter whether you're talking about tropical or temperate. It's just a piece of wood. Some pieces of wood are very easily recognized as the beech and the oak and the ash, as we're talking about. Purple heart, which I showed you in my talk, is a purple color, so that's a very good start. If you've got aliform and confluent parenchyma and you've got marginal parenchyma and you know the thing comes from Africa, then something like Afcelia is a possibility. If it comes from Asia, it's possibly Insia, which looks very similar. So it partly depends on your experience. And of course, people say in Brazil, they will have quite a bit of familiarity with the Brazilian trees that they encounter in trade there. People in Asia, the same, things from there, Africa from there. But if you're in a place like Britain, which is sucking in timber from here, there, and everywhere, where you don't know where it came from in the first place, there's an awful lot of, in supply chains, timber felled in Peru, transported over to Southeast Asia and then maybe China, turned into a product and then exported somewhere else like Europe or North America, then answering the question, what is it and where it comes from are two very much more complicated questions.
So if I want to conclude, can I say that to be on the safe side, it is necessary to do some microscopy analysis? I mean, take thin section, and not always microscopy feature can help us. And if you want to be sure,
well, I think microscopy will get you a very long way quite often, but there are certain taxa which are very tricky. Like you could cut sections of a piece of tropical wood and tell that it's a member of the family Lauraceae, but then you've got your work cut out trying to work out which genus it might be, and forget it for species if you don't know where it came from. I mean, the answer to your question really is it depends, and it depends on how much background information you have as to how far you can go.
Okay, thank you.
So apologize, I would like to interrupt here. We are approaching the end of this workshop. I suggest Peter and Reza, if possible, you interact also bilaterally, maybe later on to resolve. I also see Rosanna still has a question, but apologies, we are running out of time and I would like to be in time, being Swiss, as Angela mentioned. I don't want to be flexible about the end. Okay, so thanks for that. Good, so I would like to share a screen with a small wrap up from my side. So the speakers provided us their final slides beforehand, which allowed us to try to summarize. And here is a summary, forgive me if it's not complete, but in terms of open questions and wood identification, well, we realize here that, okay, there is potential to also base them on quantitative wood anatomy. So maybe I try to also add the laser pointer, then it's easier for you to follow here, right? Then one of the many ever occurring open questions was about plasticity of quantitative wood anatomical traits related to phylogeny, environment, and ontogeny. I think there were talks to each of these aspects, if you remember. But then also standardization, of course, is very important. So we cannot always compare raw values, that's also a point Peter Heath made, for example, when it was about pooling data, to be careful, but also others mentioned that. And a last open point was about functional role of quantitative food and anatomical traits. So we also need to understand basically what do they mean. We want to go beyond just describing the structures. We want to really understand, well, how is it related to tree functioning and also resilience to climate change. In terms of research gaps, of course, they are quite related to the open questions. So we have seen and heard we need more sampling, and this applies to everything. So obviously it's not enough to give an example to just have one piece of wood analyzed for a given species. We need more replication. Then we also need to understand what are the key anatomical traits for modeling, for vegetation dynamics, for example, but also modeling in general. I think this is very important, of course, when we want to use such microscopic information for upscaling and by this also gain more relevance. Then a point that was raised more than once was about the metadata. So please, when you collect anatomical data, also think about collecting enough metadata. One thing here specifically was a tree height, right? That was mentioned more than once. And then another point was about this trait coordination, or as I phrase here, integrative studies that place quantitative food and anatomical data in the context of other tree characteristics. Otherwise, we might draw the wrong conclusions. And then finally, well, this was also raised that the protocols and tools should be further improved to make it easier to collect more data, to also facilitate data production in general. This was also raised a couple of times. So this is a bit how we saw it in the coordination team. And well, then maybe a few last words. So I think you all agree it was a very fantastic and rich and diverse workshop today, in which we learned a lot, and probably not everything is answered, could be answered. Maybe there is more questions being raised or popping up. And so please answer them to the QNet forums. There is a forum which just waits for your question, and I'm very sure there will be someone to reply. So please use the forum functionality of the QNet webpage. We will for sure also organize a next workshop in about 6 months again. So we keep this rhythm. For this, we are eager to learn from you to what are your suggestions. So what topics should it be about? Maybe also the format. It should be interactive. This is the idea. So please interact with us, with each other, with the various ways that are also listed here. And before we really close, I would like to hand over very briefly to Achim Breuning for an announcement. This is now not in a forum, but maybe will be placed there as well. So please, Achim.
Thanks, Georg, for giving me the word again. This is just a short advertisement. We are organizing the TRACE 2022 in my laboratory next year, end of May, if Corona conditions will allow, of course, but we didn't lose optimism that after this winter, hopefully the situation will again and hopefully finally improve. We have not yet made the final program. We'll discuss it this week with the ATR board, but I hope that during the first week of December, the homepage can go online, but I promise you there will be definitely some session related to quantitative wood anatomy issues as well. So you are warmly invited, hopefully, to join TRACE 2022 in Southeast Germany, Bavaria. Details will follow on the webpage. Thanks a lot.
Thank you very much, Achim. And this brings us to the very end. So today we were overall a maximum 95 people, if I counted correctly. I think this is a very nice number, and most of them stayed quite late, which is not a given considering the different time zones. So I would like to thank, last but not least, here to the presenters. I think you agree with me, they all did a very nice job into making this a very interesting program and seminar. And then I also would like to thank everyone for participating and contributing to the discussions. Thank you very much. And the recordings of this seminar or workshop will be made available in due time. So just wait and see. Thank you very much. And maybe I stop sharing so that maybe everyone can show his face and we can hail and then, well, close this meeting. Huh? Okay. So bye-bye, everyone. Looking forward to see you very soon again. Bye-bye. Thank you.
Bye. Thanks.